presentation is minimize harms, maximize the benefits for cancer cure. And in that, I have added also that the cancer cure can be achieved or accomplished by hitting the cancer weaknesses instead of going for the strength of the cancer cells or the cancer disease. So in that context, I would like to introduce that the harms, I mean only the trauma and the psychological stress that patient undergoes is a type of harms. And the harms also include about the disease-treating drugs. So how the disease-treating drugs affect the whole physiology of the patient is very important. That is included in the harms portion. And in the first slide, I would like to say that any cancer cell, before it becomes malignant, functionally as well as structurally, the carcinogenesis process undergoes that 360-degree circle, and it involves the cancer cells in vivo as well as the ex vivo changes. In the in vivo changes, the exogenous and the endogenous factors, which are mutagenic, can able to cause the mutations. In addition to that, some growth factors from the extraneous sources can also induce the cancer cell transformation. Also, excessive hormones, especially for the reproductive cancers, the excessive hormones. In the case of breast cancer, especially the postmenopausal women, the excess secretion of the estrogen is responsible for the transformation of the normal cell into a cancer cell. And with regard to the ex vivo conditions, again, the nutritional pattern or the nutritions are very important contributor for the transformation of the cancer cell into a malignant cell. In that context, the sugar, then the essential fatty acids category of omega-6 category, and the cholesterol derived from the red meat are the inducing factors. And the normal cell, when undergoing the transformation, it takes a very long latent period of 15 to 20 years. Then why such a long, prolonged latent period means that it prepares for the transformation as well as for the functionality of the malignant cells. And recently in 2011, Perucha et al. Have revealed that the cancer cell has got the mutations, the extent of about 1 lakh. And beyond 2011, in 2016, some researchers have revealed that the mutations may go up to 160,000 mutations. How come this is possible? Means we have got so much of environmental factors as well as the health factors and the nutritional patterns of the individuals that are all contributing for the mutations. As a result, the culmination of mutations and synthesis of the various oncogenic proteins, various oncogenic receptors, as well as the various proteins and other growth factors are representing 180 degree. And when it comes to the proteins as well as the receptors will come and overlay over the cellular membrane. So the transformation involves a vital property, normal cell, namely the cell membrane. So the cell membrane already in 1960s, Rashi and Minsky revealed that the cell membrane glycocalyx composition in a normal cell includes the lipoproteins, glycoproteins, and the proteoglycans in a specific ratio. But this ratio is completely modified in the case of the cancer cell or after the oncogenesis. And these glycocalyces are responsible for the intake of the various kinds of substances which promote which are pro-carcinogenic in action. The next slide, already we know that for the cancer treatment, three principal modalities of treatment are existing now, and the World War II is the beginning of chemotherapy era for the cancer treatment. And the mustard gas or the yellow mustard gas survivors revealed the decrease in their WBCs, and that gave a clue that it can be employed for the hematological cancers like leukemia. And there arises the first chemotherapeutic drug against the hematological cancers. And no doubt, I am very much sure that any cancer cannot be cured without the chemotherapy, because the chemotherapy involves different types of drugs, anti-cancer drugs, which are of different kinds, like antimetabolites, biological response modifiers, and then antibiotics, then alkylating agents, etc., etc. And what is the result means the chemotherapy brings about 85% of the cytotoxic killing. And what is the sad part of this chemotherapeutic treatment is in the solid tumors, the 15% of the residual cancer cells will remain alive even after the chemotherapeutic treatment. And during the remission period, after the remission period is over, these 15% of residual cells which survive the target therapeutic attack of chemotherapy will revamp themselves to resuscitate or to reform the tumor at the primary site itself. And they become more resistant also to the radiation as well as the chemotherapy further, and they are able to metastasize the body. That is the beauty of this 15% residual cells, which includes the following cells in the tumor area, primary sites. That is, one is a resistant cells due to the chemotherapeutic attack and which survive, and the stem cells. So this is very important. All tissues have got the stem cells like this. The cancer tissue also have got the stem cells. And according to recent literature, 15 to 20% of the stem cells are present in various, depending upon the organs which are made into a cancerous tissue. And in addition to that, they have got embryonically undifferentiated cells. So these undifferentiated cells are responsible for the secretion of certain growth factors, as well as the placental hormones, as well as the antigens, and also the basic carcinogenic antigens and all. As a result, this 15% becomes responsible for the tumor recurrence. And I already told that the cancer cure as well as prevention remains in the nutritional uptake only. So in 1990s, the American publications and journals have written about the importance of nutrition. That is, for the cancer patients, the nutritional pattern is widely neglected, and the articles also stress the global level nutritional pattern, and also the nutritional support in the cancer patient, and about the protein nutrition. And also they have mentioned about the protein nutrition, and also how far the nutrients or the nutrition will become a prognostic value for the diagnosis of the cancer. So in that way, they have pointed out the nutrition of the dietetics for the cancer disease. But the various agencies which are involved in the chemotherapeutic research or the therapeutic research are brushing aside the nutritional dietetics pattern as evidences, as clinical evidences are lacking for these materials. In India and all, and in Asia, before 3,000 years or 4,000 years back, the vegetation, the various herbal constituents and the herbal parts, as well as recently, our own students have done some work on macroalgae in the marine ecosystem. So, comprising both the terrestrial domain as well as the marine ecosystem, we have found out that different types of plant materials have got anti-cancer properties. Not only that, their anti-cancer properties depend upon the antioxidant property, and then their antimicrobial property, and also their cytotoxic killing. And these, I always mention that the cancer cells derive the glucose. When you supply glucose, that is why I often stress that for the cancer patients, they should not be advised to take the simple carbohydrates. The adaptation to get another material, that is the amino acid glutamine, that will get an upheaval. As a result, the glutamine will also support the survival of the cancer cells. When once this glutamine is also down, then another amino acid, namely the asparagine, will take part, and that will come to the cancer cells survival, or it will make the cancer cells viable. So this it is not only involving the carbohydrates, but the proteins and amino acids also contribute to the hormone metabolism of the cancer cells in the form of glutamine or asparagine. And we have got also different kinds of amino acids. And if you see in my work, in my students' work on E-cadherin gene, in the E-cadherin gene, we have seen that the branching chain amino acids pattern is completely variable when compared to the non-tumorous region. And we have found out the ratio between leucine and isoleucine as the control it is 3:2, whereas in the cancer tissues like uterus or cervix or the breast or in the colorectal sites, it is 3:4 or 3:5 it goes. And likewise, we have taken 200 base pairs, both the forward reaction and backward reaction. And in that, the complete normal sequence have been completely altered, just like a frame shift mutation. Just like a frame shift mutation, the mutations. So this justifies the statement of Peruto et al. That it may take about 1 lakh and above the mutations to culminate in the form of a cancer cell. So when you come to the lipid side, the lipogenesis is also one of the bad effects of cancer cells. And in the lipids, you have got two types of essential fatty acids. One is omega-3 and omega-6. And we have found out that in all cancer tissues, the omega-6 EFAs are essential fatty acids like linoleic acid, linoleic, and then myristic acid, then lauric acid, then palmitic acid. They are all in the high proportions when compared to the omega-3 essential fatty acids, especially the alpha-linolenic acid. So the linolenic acid is anti-cancer in effect, whereas the other essential fatty acid which I mentioned is responsible for the pronunciation or the promotion of cancer growth. So when you restrict the supply of omega-6, especially in USA, I have read in one book or reference that if the ratio between the 3 and 6 is 1:1, it's very good. Whereas in a normal person, it goes to omega-3 1:2, omega-6 20. And in the developed countries where the fast food centers are in the sway, they have got about 1:50 ratio of 3:6 omega essential fatty acids. And so that is why the USA is the capital of colorectal cancer as well as the breast cancer diseases. So this omega-6 fatty acids in our study, they have taken the gas chromatographic analysis and found the clear-cut obvious differences between the good essential fatty acids. That is why I have mentioned that the omega-6, omega-3 and cholesterol. Omega-3 is the good, the omega-6 is the bad, and cholesterol is the ugly form of the lipid materials. And in the weak point number 3, all cancer cells niche is acidified. And already you know very well that in 1931 or 33, the Warburg's concept is, as mentioned, that the tumor microenvironment is acidic because they synthesize by fermentation. They undergo the fermentation reaction of that is why they cannot able to survive. So when you change this acidified environmental micro niche against surrounding the tumor environment, namely in the cells, in vivo and ex vivo as well as in the stromal cells, then we can able to control the growth of the tumors. In that context, I would like to mention that so much of plant materials, fruits and seeds are responsible for the alkalinization of your body and also your redox balance. And as a result of alkalinization, even in China, recently they have found out that bitter gourd water, hot water is bringing about the alkalinization. Likewise, the coconut water, hot water, bringing about the alkalinization. Likewise, the pineapple hot water is responsible for alkalinization. If the tumor microenvironment becomes acidified, it will make the neighboring cells, contiguous cells also tumorous, and at the same time, sometimes the OH radicals will come out of the cancer cells, and nearby fibroblast cells also will become the cancer. So that is why in certain breast cancer treatment and all, the victim or the patient, when they are given the chemotherapeutic drugs, in addition to their normal side effects, sometimes they also suffer due to hematological cancers like leukemia. And so this alkalinization is the weak point number 3. And the next one is the oxygen therapy, which is practiced in USA. That is in a hypoxic environment, the cancer cells survive. Whereas in a hyperoxic environment, it is not able to survive. This is also an ambiguous statement as far as the oxygen content is concerned. The tumor itself will reveal 3 zones. One is a peripheral zone and a sandwich intermediate zone and the inner core. Inner core is completely free of cancer cells. The middle sandwich layer is sparse cancer cells, and peripheral layer only is crowded with more number of cancer cells. So the hyperoxic environment and the oxygen therapy is also being carried out in US and other European countries. That becomes a weak point number 4. So you have to reduce the growth of the cancers by supplying more oxygen. That is an ambiguous point to be verified by all people. And the next one, and this is the cancer weak point number 5, that is the oxidative stress. So the oxidative stress in cancer cells will produce more amount of hydrogen peroxide. When the more amount of threshold limit of hydrogen peroxide exceeds, then the cancer cells themselves will die. As a result only, these cancer cells will convert this hydrogen peroxide into OH radicals, and these OH free radicals will diffuse out. As a result, they will come to the stromal cells and they will convert the stromal cells and the fibroblast cells into cancerous tissues. Can you follow? So that is why this planning is very important. How to control this OH, or how to increase the OH in the cancer microenvironmental mix is very important as a therapeutic measure. And finally, I come to my topic that nowadays so much of herbal nutrients and the herbal components have been identified. All will have the anti-cancer effects. We have ourselves tried in different plant materials like Vitex negunda, one species, then Carica papaya, the papaya fruits and the plant materials, the leaves and all. And then there is another one is called the neem tree, as well as the latex of calotropis. My students have worked on this using the cell lines MCF7. The most aggressive breast cancer cells are being employed for these tests, employing the NPT assay and the dye binding assay, that is Python blue, the binding assay. They have all revealed that these plant bioactive compounds like flavonoids, then flavones, then beta carotenes or carotenoids, then the triterpenes, and then the carbohydrates, and then the sterols like beta cytosterol and phytosterols. One important message about the phytosterols means it is much prevalent in the fruits when they are in the unripe condition. So in the unripe condition, the phytosterol and beta cytosterols are very much present in the unripe fruits. And one of the method of reducing your prostate cancer, which is also a hormone responsive cancer, and one of the methods of herbal treatment is to take these fruits in unripe condition, which is full of the beta carotene, the cytosterol and the steroids, and they will just like tamoxifen, which is acting against the breast cancer cells. These phytosterols and beta cytosterols will block by competitive inhibition. As a result, the breast cancer cells can be prevented from intensive proliferation or proliferation. So that way, we have analyzed the different species of plants, and we have also come to the next one domain, that is the marine domain. So in the marine ecosystem, the macroalgae constitute a very important source of materials which are anti-cancerous in nature. And in that, my students have worked on Carbonaria, that is one algae, that is brown algae. One is a Sargassum myriosystem, another one is a Carbonaria ornata. In both these researchers, they have phytochemicals identified, and they have also employed the ferrous nanoparticles, and another one employed the another zinc nanoparticles, and found out that these nanoparticles act as chaperones for these phytochemical molecules. As a result, the target therapy becomes easy, and the nanoparticles enter into the cancer cells immediately by penetrating the cellular membrane, and they are responsible for the sustained release of these compounds which are anticancerous, especially the phytachinoids, then saponins. They have found out that the saponins are membranolytic in action. And so when you apply the saponins as the mitochondrial release of calcium to the cytoplasm is enabled. As a result, it will automatically go into the caspase-3 action, that is the apoptotic main enzyme responsible for the programmed synthesis of the cancer cells of the apoptosis. So with this, I come to the conclusion that the marine ecosystem contributes so much of non-edible varieties of algae, and which are still remained as non-conventional food items only in Asiatic countries, particularly in India. And likewise, so much of vegetables like that broccoli is now coming up. My insistence. They have made it as a table menu daily. Likewise, this button mushroom and oyster mushrooms are very much important because they contain the lentinan, and the broccoli contains the anti-cancer compound, the sulforaphan. Likewise, we have identified different composition of the different herbal constituents as well as the marine algae. And these marine algae, especially the fucoidin, the sulfated polysaccharides present in the algae, marine algae like Turgeneria or in the sargassum, they are responsible for the bringing down this DNA fragmentation, bringing up the DNA fragmentation as well as the programmed cell death. And the saponins will break the membrane lysis. And I insist upon taking the Budwig's diet, which was taken by a lady doctor, Dr. Budwig. And he gave to the myostatic patients the cheese plus herd plus flaxseed oil. Now we can use, instead of flaxseed oil, the chia seed oil also. And that will be the diet of the cancer patients in the lunch. And likewise, the green tea and the white tea, which are very heavily antioxidants. They can also save the tissues, not only save the normal tissues, but they can also pinch the free radicals formed inside the cancer cells. And sometimes the olive oil, the cooking oil should not be the vegetable oils, usually the vegetable oils instead of palmitic oil. I have already given in my lectures that the palmitic acid is one of the culprits for the carcinogenesis process. So instead of using the palm oil and other sapola and canola, you can go for the coconut oil or the olive oil or the flaxseed oil in the cooking purposes, and that will be a solace to the cancer suffering patients. So we have all got a life to live and not to die due to cancer, and it is not the God's destiny. So with this, I thank you very much for giving me, and I very much thank the Luna Watkins, and she only introduced me to this talk. I am very much grateful to her, and I also thank on behalf of the Dubai people and the Dubai organizers that it is a conceptual finding only. We have also done some glutamic oxaloacetate transaminase reactions. And these transaminases are a good marker of the for the cancer process. And in all my studies, I have presented all the results. Even in the recent paper, I have read that the cancer cells undergo the oxidative phosphorylation. How come I question? How come it be possible? Because we have seen the normal architecture of the mitochondria has been altered in the cancer cells. We have got an ultrastructural structures of the cancer cells mitochondria in the uterine cells or the cervical cells or in the breast cancer cells. And I suggest that once the oxidative phosphorylation is probably partially may be occurring, and full TCA cycle will never be expected to take place in the cancer cells. And in parasites, the partial TCA cycle is operating. It is already well known by the people, by the scientists. And likewise, the microbial organisms depend upon the fermentation reactions for the energy. So these cancer cells go back to the primitiveness, and as a result, they stimulate the metabolism of the primitive organisms like the bacteria as well as the endoparasitic species, and as a result, they derive the energy for both their anabolism and catabolism. And that is enough because once a cancer cell is formed, it can able to absorb 84% of the glucose, total glucose available in the body, whereas a normal cell takes only 14 to 16% of the glucose. Think of it. So with this 5 times increase of this glucose supply, the cancer cells by fermentation can able to bring about an environmental niche which is acidic both interior as well as exterior, and it is able to synthesize the oncogenic proteins and receptors which cover the surface layers. And when you take red meat, and the cholesterol intake is responsible for the formation of the caveolae. The structure, already I said, the cellular membrane structure is altered. The caveolae is a bit-like structure in the cancer cell membrane, where the cholesterol layers are arranged in such a way that they promote the glucose absorption very efficiently. And accordingly, the inner lying glucose transporters, there are 13 different glucose transporters are available. We have got studies with regard to the glucose transporter 4 and glucose transporter 1 with regard to the cancer cells, and they are released to the membrane, and they've been able to transport these glucose recombinantly, and this will be the adaptation. That is the adaptation of these cancer cells. And already I said that the proteins, proteinaceous nutrition, they have neglected, but the protein intake of too much of proteinaceous food is also dangerous, because we have already seen the implications of the proline. When the proline is converted into hydroxyproline, it will suppress your immune system. I think the speaker who mentioned about the CD8 cells which come to the rescue of the cancer patient, whether they will be functional in the environment of the hydroxyproline, and whether they will be functional in the case of the glutamine-rich cancer cells, whether they will be able to function in the microenvironmental niche which is acidic in pH. All these things and other ramifications should be brought into the field. And I have written the titled book, 2 personal publications of mine. One is Proven Proxies to Prevent Cancer, and the second volume is again the Proven Proxies to Cancer. In that I have mentioned all types of herbal terrestrial herbs, and including the vegetables, seeds, nuts, oils, and even the spinach. The fin is in Canada research, they have found out that the only tissue which is free from cancer is your cartilage. And the cartilage factor they have worked out in Canada, and some scientists have proved that this has got an anti-angiogenic property in the fins. And I have also suggested one fellow who suffered the nasopharyngeal cancer, and he started taking this fin soup daily, and he is now free from all the side effects of nausea and headache, and he is now completely cured, and he has joined the police job. So that much beauty lies in our natural elixirs of both the terrestrial ecosystem as well as the marine ecosystem. So thank you for giving me a chance to mention about the phytotherapy and its implications to cancer. Thank you.
Great. I would
thank you for all for arranging this nice meeting. And this meeting actually mainly we are observing these are mainly associated with the modification of different type of cancers and other epigenetic other mechanism. But the topic which I have chosen is totally different, because till now we knew that cancer is the origination from the genetic perspective. But now we are the first to show that epigenetic control of T helper cell differentiation, how it can controlled by the nucleotide excision repair proteins. And that is why one of the major nucleotide excision repair protein is the XPG, that is the Xeroderma pigmentosum complementation group G. So here we can see that this particular protein, when they are mainly associated with the NER pathway, and they are actually targeting the NER pathway via the different type of tumor suppressor protein, especially the BRCA1. And why we have chosen the T helper cell, that is the question here, very interesting, that which we have shown that mainly it has in the recent time, this T helper cell showed that it produces effector cytokines such as IFN gamma, TNF alpha, which have direct anti-tumor activity, and following the activation and polarization into Th1 phenotype. As you all know, the T helper cell has different phenotype like Th1, Th2, Th17, Treg, all these things. And now among this Th1 phenotype, which is mainly respond to the signals from the dendritic cell, particularly via one of the interleukins that is known as IL-12 or interleukin 12, which actually induces the expression of the particular components of the nucleotide excision repair components associated with XPG, and which actually mainly shown in the case of breast cancer, which patient we have got, they are deficient of this XPG expression. And it has already been established that the epigenetic regulation plays the major crucial role in T helper cell differentiation by other lab and our lab and other lab. And thus, we can hypothesize that possibly this XPG is mainly involved in the epigenetic regulation of the T helper cell differentiation in the breast cancer. So next, we have chosen some methodology where we have, with the ethical clearance from our institutional ethics committee, that we have collected some blood from the patient who are suffering from IDC, that is the invasive ductal carcinoma, and then we have isolated the CD4 T helper cell using the MAX technology, and that MAX technology, then we have taken this particular CD4 T helper cell, and then we go for the CRISPR-Cas9 mediated XPG knockout and overexpression study, and then we have chosen those expressive this XPG knockout and overexpressed cells for the rest of the study or the downstream processing of the study regarding say as RNA isolation, then ELISA, then we have cytotoxicity assay, then we have RT-PCR for the gene expression, then we have done the chromatin immunoprecipitation to see the enrichment of that particular factors which are associated with that particular T helper cell differentiation factors. And also we have seen this how this suspension of this cell fixation are mainly associated with this particular breast cancer etiology. And that's why when we are choosing this parameter, we have chosen one of the very interesting phenomenon that is known as M6A RNA methylation, which is also a kind of epitranscriptomic mechanism. So epigenetic plus epitranscriptomic both are actually going hand in hand with the help of this XPG protein. So now we are coming to the first slides where we are showing this RT-qPCR quantitation of the candidate genes, which are mainly involved in the T helper cell differentiation in unskewed condition. That means in the naive T helper cell population. In the naive T helper cell population, which we can see here that if you compare the fold change normal over the breast cancer, it is showing IFN gamma, TVX21 are high, but instead of that, IL-4, GATA3, IL-10, FOXP3, IL-17A, ROD gamma T, they are very low. So that means this mRNA expression profile are clearly indicating that in the normal scenario, the Th1 specific cytokines and the transcription factors are highly expressive in condition to, in respect to breast cancer patient. But the opposite effect we have seen in case of IL-4, GATA3, that is the Th2 specific cytokine and transcription factor, IL-10, FOXP3, that is the Treg specific transcription factor and cytokine, and the Th17 specific cytokines, that is the IL-17A and the transcription factor ROR gamma T, they are very upregulated in case of breast cancer. So this actually triggers us to choose how we can go for this particular stage 1 breast cancer patient. And then what we have done, we did a very simple experiment. We have first isolated the blood, and from the blood, we have isolated the particular the cells, and then we have go for the first we have through the CRISPR-Cas9 we have actually knock out that particular XPG, and also in another system we have actually overexpressed the XPG. And now you can see in this particular construct which we have used, it is the CRISPR-Cas9 construct which has the GFP as a reporter, and you can see everywhere GFP expression is there. But when we are actually knocking out the XPG, you can see the graph half of the facts, it is going in the left side in the case of knockout. And again, if you overexpress, then you can see it is moving towards the right side. That means this particular overexpression and knockout we have successfully done, and we have also found out that how this particular expression of the XPG are actually regulated via the different kinds of DNA repair and DNA repair and breaking pathway like XPG, gamma H2A.X, P53BP1, and there we are seeing that in case of control knockout and the XPG knockout in comparison to control, what is the status of them in the case of breast cancer. And now you can see in case of XPG knockout, the XPG is nothing there, but in case of gamma H2X, it is highly expressive in both control knockout and also in the breast cancer patient. That means this gamma H2X actually represent the double strand DNA break, which is mainly associated with the phosphorylation of the histone H2A.X protein. And also we have seen that 53BP1 is also expressive highly in case of control in comparison to breast cancer patient. So next, we have shown some RT-qPCR data, which shows the downregulation of the cytokines and transcription factors, which are mainly associated with the T helper cell differentiation, in especially TH1, like TBX21 and also IFN gamma, which are mainly downregulated in case of CRISPR-Cas9 knockout system, but it is upregulated when we overexpress that thing. And also we have seen the IL-4, GATA3, IL-10, IL-17A, rod gamma T, they are highly expressive when we are actually knocking out the XPG, and a reverse effect we have seen when we have overexpressed the XPG. That means the XPG playing a crucial role in modulating the T helper cell towards the TH1ness and diminishing the TH2ness and TH17ness and Tregness. And also we have found out in the next slide where we are showing this RT-qPCR quantitation of the candidate genes, which are mainly involved in the DNA repair and tumor progression at unskewed condition. That means in TH0 condition, where it is showing that H2FX, ATM, TP53, BRCA1, all are highly expressive when we compare with the normal over breast cancer. And similarly, we have also taken a look on the H2AFX, where we have seen that normal control XPG, what is the status, and when we are actually doing the XPG knockout, what is the status. So these status are actually indicating that particularly the fold chain of this TH1 over TH0 condition and TH2 over TH0 condition, TH1 over TH17 condition, all these things, and they are mainly associated with the knocking out of the XPG, mainly resulted in the downregulation of the genes, which are mainly involved in the DNA repair like H2FX, ATM, and the tumor suppression like TP53, BRCA1 in terms of fold change, that is TH1 over TH0, and normalized to GAPDH gene for T cell, for which initially isolated as the CD4 T helper cell from the PBMC of 3 control subject and 3 breast cancer patient suffering from stage 1 cancer using the max cell separation technology. Next slide, we are showing that how this, okay, if it is the scenario, so what are the factors mainly associated with this XPG? So that's why we did a cheap qPCR assay, that is the chromatin immunoprecipitation assay. That you all know, chromatin immunoprecipitation, nothing but first you fix the cells with the paraformaldehyde, and then you are going with the particular antibody and immunoprecipitate that particular segment of that particular sequence. And there we have found out in case of XPG cheap on the IFN gamma gene locus, in the case of normal control knockout, it is highly expressive, but when we are doing XPG knockout, it is highly diminishing. Similar kind we have found out in the ERCC1 chip, that on the IFN gamma gene locus, it is showing in the normal control condition, control knockout condition, and high expression of that particular enrichment of that particular factors, but it is low when we are doing in the XPG knockout. Similarly, we have seen the another DNA repair factor, that is the CSA chip on the IFN gamma gene, and it is showing the high enrichment in the normal control knockout, but it is diminishing in the XPG knockout condition. And also we have seen, interestingly, the reverse effect with the gamma H2A.X chip, and this is mainly associated with DNA double strand break, and it is showing when XPG is knocked out, that time the enrichment of the gamma H2X is high in case of both normal as well as in the breast cancer patient. And also we have seen that what is the interrelationship between the chromatin factor, that is only CTCF, which is mainly associated with looping up between the different type of similar kind of genes. And here we have seen in case of normal control knockout is highly expressive, but in case of XPG knockout and breast cancer patient knockout as well as control knockout, all are diminishing. That means the reduced percentage enrichment of the XPG and its associated NER complex actually forming partners like CSA, ERCC1, but increased cluster enrichment of the DNA double-strand break marker gamma H2A.X on IFN gamma locus in XPG knockout T helper 1 cell and CCTC binding factor, that is CTCF, and XPG actually orchestrated this TH1 cell specific chromosome structure and function which were observed in the IFN gamma gene locus. Next, we have shown that how this cheap qPCR increased the percentage enrichment of the histone-associated altered modification, where HDAC3 corresponding to histone deacetylation and decreased percent enrichment of the H3K4 trimethylation and increased percent enrichment of the H3K9 trimethylation corresponding to histone demethylation was observed from the data of the XPG knockout T helper 1 cell of breast cancer patient, and uniform percent enrichment of histone H3 at IFN gamma gene locus indicated that the chromatin quality was maintained in all throughout the condition. So next slide, we are showing that how this, if this is the situation, what is the status of the epitranscriptomic mechanism? That means what is the status of the M6A RNA methylation state? And it is showing the colorimetric assay showing that reduced amount of the M6A RNA methylation observed in the XPG knockout and T helper 0 cells compared to control knockout and TH1 cells. And also we have seen the similar kind of effect in the case of extracellular secretion of IFN gamma and other thing where XPG is knocked out, then IFN gamma release is reduced. But in case of IL-4, the IL-4 release is exceeds when the XPG knockout is there. So next, we have taken a look whether if it is the case, whether this is helpful for killing the breast cancer cells or not. Yes, we have found out through our LDH release assay study that the percent cytotoxicity of the CTL, which is mainly triggered by the T helper 1 cell differentiating factor IFN gamma to trigger the cytotoxic T lymphocyte, which observed to be reduced in the XPG knockout in comparing to control knockout for both control donors and breast cancer patients PBMC on interacting with the MCF7 breast cancer cells. And also we have found out that how this RT-PCR, qPCR actually knocking out is helping in the condition of the different type of factors which are associated with the mainly the apoptotic pathway, and it has observed that it is also increased in case of anti-apoptotic pathway, which increased in case of XPG knockout. So overall studies actually showed, this is the pictorial representation where we are finding that if the XPG is present, then TH1ness is increased, TH2ness, TH17ness, T-regness is decreased, and that actually triggers the TH1 cells to release the IFN gamma, which will increase the, which will trigger the CTL to generate the cytotoxic T lymphocyte, which will kill the breast cancer cells. And at the same time, we have observed in the case of XPG presence, it gives us the protective tumor protective immunity. In case of XPG absence, it causes the immune deficiency and genomic instability via the T helper 1ness decreasing and T helper 2ness and TH17ness, T-regness is highly upregulated. And at the same time, we observe that in case of XPG absence, the histone deacetylation is increased, histone demethylation is increased, chromatin looping is decreased with the in the case of CTCF, and also DNA damage repair factors are decreased. So this is the overall pictorial presentation of the whole talk. That means the XPG playing the major crucial role, not only increasing the TH1ness, decreasing the TH2ness, but also it regulates the epitranscriptomic as well as epigenetic mechanism via the regulating the histone acetylation, histone methylation, and M6A RNA methylation. So this is our whole objective, and this the 3% in the triangle, they actually have done the work for my lab, and they are Riyasha Pal, Nilanjan Paul, and Deep Bhattacharya. And we are really thankful for our SRM for giving us this wonderful facility. And also I would like to thank our SARB DST, Government of India, for financial support for this project. Thank you all over here, and I am now ready for a question.
Consultant medical oncologist, HCG Bangalore. So This is one of the old paper which we have done a study, a retrospective study, where around 60 to 65 patients we have analyzed who are taking a trastuzumab injection for both locally advanced and metastatic breast cancer patients. Though at present point of time we have so many anti-HER2 therapies are available, including pertuzumab, TDM1, and now this N-HER2 therapy, trastuzumab, deruxtecan. So this paper at present point of time may not be relevant, but during, I mean, it published in 2020, 3 years back. So during that time, there were more biosimilar coming up, and still not many newer therapies, newer anti-HER2 therapies were not available at that point of time. So we just made a effort to see the response rates with our center, as well as we wanted to see the different biosimilar compared to the original, the response rates and survival rate with the biosimilar and the original. That is what we have done in this paper. That effort we have done to do in this paper. So basically it is like going back to revisiting our older version of treatment for metastatic breast cancer and locally advanced breast cancer. Though this present form of therapy, most of the for locally advanced, we go ahead with either the pertuzumab trastuzumab combination along with chemotherapy. So that is what, but now that is that is not there in the, I mean, only trastuzumab with chemotherapy is considered to be a suboptimal. So before going to that, you can see in the right side corner of my slides, it's a time, mom, it's time for mammogram. So I am on a mission to impact 10 million women to detect the breast cancer early by doing the mammogram. Screening mammogram awareness in India especially is very low, and most of the breast cancers detected in India is almost in advanced stages. So screening mammogram detected breast cancers may be less than 1%. So that is why I felt there is a necessity to create awareness for screening mammogram for our people in India, so that we can detect the breast cancer early and cure it. So that is why in this this campaign with a logo with a tagline of "Mom, it's time for mammogram." So coming back to the scientific study, so this is the published paper, clinical profile of carcinoma breast patients treated with trastuzumab, a single center study, and it published in advanced scientific research publishing paper, Advances in Breast Cancer Research in 2020. So this is the introduction. Breast cancer is the most common female cancer in India, accounting for almost 1 in 4 cancer cases in women worldwide. And according to Globacon 2018, so breast cancer incidence is increased to 1,62,000 in 2018 and compared to 1,44,000. And now it is keep on increasing. Biosimilar drugs allow expanding access to the therapies in the form of cost savings and leading to better overall health outcomes. So many biosimilars have come up in trastuzumab, and in the TDM1 also we have a newer biosimilar which we are using more frequently than the original because of the lesser cost. So we don't know at that point of time also we don't know, and now also we don't know. I mean, though there some studies are there where they compare original and the biosimilars, but we feel that the robust studies comparing head-to-head comparison may not be there. So our study evaluated the efficacy and safety of trastuzumab biosimilar and assess overall survival in the study population. So objective of the study was to assess the response rate for trastuzumab in metastatic and non-metastatic breast cancer patients, to evaluate overall survival and toxicity in these patients. So this prospective study was conducted in healthcare, our health HCG hospital, Bangalore, India. And all female patients diagnosed with HER2 positive metastatic MBC and locally advanced breast cancer between March 2013 and November 2014, with at least 4 years of post-treatment follow-up were considered for this study. So ER positive, PR receptor, ER receptor, HER2 receptor, K67 was done, by IHC, and HER2 status, if it is 2+ by IHC, then we would like to, we have done a FISH testing to consider it is a FISH positive. And hormone receptor status and HER2 positivity were confirmed before initiating the chemotherapy, and only patients who followed standard guidelines for trastuzumab, Herclone, Biceltis, and Canumab were included in this study. So patients received first dose of trastuzumab 8 mg per kg, and subsequently every 21 days received as a dose of 6 mg per kg for almost 17 cycles. All patients underwent the imaging study, including CT, MRI, PET CT, depending upon their metastatic status, before and after treatment with trastuzumab were assessed, and the response rates were assessed for partial response, stable disease, and progressive disease. ECG, echo to assess the cardiac toxicity with the ejection fraction were recorded before starting and after 3 to 6 cycles of therapy. Toxicity was considered when ejection fraction dropped by more than 10% after treatment with trastuzumab. All these clinical information extracted from the patient case files. So exclusion criteria was along with other standard exclusion criteria. Patients who are not willing to undergo treatment with trastuzumab and who failed to receive minimum 3 cycles of trastuzumab or defaulted or expired during the course of treatment were excluded from the study. So at least 3 cycles they should have completed to evaluate the response. Those patients only were included in this study. So statistical analysis as per our statistician was done. So this is the baseline characteristics of our patients. Around 65 patients we have included in this study. Mean age was around 53, 54 in both metastatic and local advanced patient. The chemo duration was the 3-weekly therapy was done. Premenopausal around 20 patients total, and in postmenopausal around 45 patients. HER2 by IHC3 was around 45 in out of 65 patients. Confirmed by FISH were 20. And then hormonal status triple positive or 31 total, and the ER/PR negative and HER2 positive were 34 patients. So chemotherapy regimen, majority of both the around 50% of the population, AC followed by Pacli plus trastuzumab, we call it as ACTH, and a TCH regimen around 35% of the patients. K67 we can see here more than 20 was almost 64, almost 95% of our patients, 97% of the patients were K67 more than 20%, and only one patient had a K67 of less than 20%. So these are the baseline, response rates in our study. The overall response in metastatic patients with any of the regimen, ACTH or TCH regimen, was 46.1%, and stable disease was 30%, and progressive disease was around 23%. In local advanced, it is a 76% response rates compared to 46% in the metastatic setting. So if we split these into different hormonal subgroups, only ER/PR negative, HER2 positive, the PR was 47%, and the local advanced stage it was around 76.5%. In triple positive, it is 44% response rate, and in local advanced it was a 77% of the patients. Compared to the generics or the original molecule, so 55% of the patient had partial response compared to 16% of the patients with the biosimilar. Though the numbers are not equivalent, so the comparison may not be that much attributable. We cannot take it as a numerically significant actually. But we try to make a comparison here whether the biosimilar or the virgin generics are, but because of the numbers we are not able to do that exercise. So toxicity wise, LV ejection fraction has less than 10%. So the toxicity was very minimal. Less than 10% drop is seen in 83% of the patients, and more than 10% of the drop was seen in around 17% of the patients. Around 11 patients had a more than 10% of the drop, and most of these patients have recovered after stopping 1 month of therapy, and then we are able to restart the therapy. So summary of the response: locally advanced patients shown better overall response, 72% compared to 47% of the patients with metastatic disease. Less progressive disease in locally advanced disease compared to the 23% in compared to the metastatic patients. About 23% of the patients in the current study achieved a pathological complete response. Those with locally advanced disease, we have done the neoadjuvant therapy. Those patients, almost 23.8% of the patients had a pathological complete response. Though many of the studies in international studies show the neoadjuvant response, neoadjuvant pathological after neoadjuvant therapy, pathological complete response is around 50 to 60% in HER2 positive patients with the pertuzumab and HER2 combination. This in our study we got it as a 23.8% of the patients, but more, not all the patients had neoadjuvant chemotherapy. Some of the patients had the surgery and then came for adjuvant chemotherapy. So that is why the response, I mean, the numbers are less and the response may also be less numerically. So response rates in virginal and biosimilar trastuzumab were not statistically significant because the number of patients receiving biosimilar trastuzumab were less. So the mean overall survival of the study patient was 20 months in metastatic and around 29 months in the locally advanced patients in this study. So the mean duration of follow-up was only 4 years. After that, we didn't follow up these patients. The main toxicity of the was trastuzumab cardiotoxicity was observed in less than 10% in 54. I mean, the toxic, I mean, EF dropping less than 10% is almost 83.1%, and 16.9% had a more than 10% drop, and we were able to restart in those group also. And apart from the neutropenia associated with other side effects associated with chemotherapy that we have not recorded, as the study was overall survival, cardiotoxicity, and the generic versus original comparison. So p value was not significant as the study population was small. So results of the current study indicate that overall survival did not differ with respect to hormone receptor status and Ki67 expression. Further, overall survival did not differ between the AC followed by taxane and Herceptin, trastuzumab combination versus the taxane, carboplatin, and Herceptin trastuzumab combination. So this study noted that patients on TCH regimen may be more likely to complete trastuzumab. However, Indian settings cost presents a significant barrier to trastuzumab in HER2 positive breast cancer patients. So although biosimilar therapies hold a promise for alleviating the cost burden on access to treatment, data on comparative efficacy and safety is relatively scarce. Results of the current study indicate that overall survival did not differ between patients on Herclone, Biceltis, and Canmab, though the numbers were less, so we couldn't draw much conclusion in this. Even response rates in virginal and biosimilar trastuzumab were not statistically significant because the number of patients receiving biosimilar trastuzumab were less. According to FDA approval on biosimilar, the response of trastuzumab with biosimilar was not superior to the generic version, as per one of a few studies. So with respect to cardiac safety of trastuzumab, it may be important to monitor ejection fraction decreases. However, as per the results of the current study, overall survival patients, we have done the survival analysis whether the more than 10% drop or less than 10% drop, any survival difference, but there is not much, no cardiac related mortality seen in this study. So this prospective study shows the effectiveness of trastuzumab for HER2 positive in locally advanced and metastatic breast cancer. The response rate, survival, and toxicity correlate with other global studies. Response and survival are same as either generic or original trastuzumab. So by this, I will conclude my study. So any questions I can happy to take up.
Operators who have been constantly working in making this event a success for both the yesterday's event and today's event. So before proceeding further, I'd just like to give brief introduction about my institute, and after that I'll be presenting about the integrating approaches in evaluating and developing the breast cancer molecular entities for effective diagnostic as well as the therapeutic approaches. I hope the slides are changing. Okay, so this is my institute where just I'm highlighting some of the blocks where our institute is one of the institute of eminence in India and of national importance, where we have an patient load of approximately 10,000 to 12,000 outpatients per on a daily basis. And this is a huge amount of burden. And these are the various blocks of our hospital and the research centers where we carry out a lot of research related to breast cancer. Before getting into the details of the actual hypothesis and the lab, what my lab does with respect to breast cancer, uh, I would like to just say that breast cancer is one of the major concerns both at the national level and even at the burden which we see in our hospital. Considering that we know that there are different types of breast cancers, uh, both the ductal, lobular, then the mixed one, both ductal and lobular, etc. Our major concern is about that what are the causes on one hand and how they have the risk associated with it. And we know that there are many kinds of uh, factors which influence the breast cancer, whether it might be the age, the emotion, the obesity, and other comorbid conditions which have a greater impact on the breast cancer. So if we look into the different kinds of subtypes of the breast cancers, I think in previous sessions, as well as the speech which was followed just before my talk, was also highlighting about the different types of the breast cancer. So I'm not getting into the details of that. But if you very carefully look, and a couple of speakers have more focus on the genetic factors. However, our lab basically looks from both the aspects of the genetic associations of breast cancer or the origins of the breast cancer, as well as the somatic. And when we talk about the somatic mutations and the genetic mutations, our focus mainly drives to classification of the somatic mutations. And these somatic mutations provide an information with time that the changes associated with the mutations provide us an information that how should be the therapeutic approach. Already we know that if it is the genetic mutations are already classified and we can see their appearance when we do the sampling. However, when it comes to somatic mutations, these are abrupt, aberrant, and sometimes they are very aggressive. And it becomes very challenging that how we can have an understanding from the diagnostic point as well as develop a therapeutic strategy. So that is the greater focus for us, that if the somatic mutations, because somatic mutations just appear in one generation and sometimes they can be getting into the generation after generation. Maybe that you might not see those kind of somatic generation in one, in one particular generation, and maybe in the second generation, you might not see them. So these are again some of the difficulties which are associated with the diagnostic approaches in identifying the biomarkers as well as developing the specific strategy. So our lab's focus was more on how we can develop an integrating approach because there are many events which causes the breast cancer. So our main focus was to develop an integrating approach, not only considering from the biomarkers identification as well as the mutations which are associated with these biomarkers. Now we know that these biomarkers have and are being influenced by mutations either genetically or at the somatic. Not only these, but the other factors such as the microRNAs play a very important role in modulating their expression. It is not just the appearance of mutation of a particular biomarker in a particular generation, but their expression regulated by the microRNAs is again an important approach in making our wider understanding of the pathological progression of the disease, as well as the other third important factor would be about the DNA repair genes. And previous talk by Sarkar has appropriately mentioned about the DNA repair mechanisms. And our focus is also, one of the focus is also about the DNA repair genes. So for the time being, and in this presentation, I would emphasize more on these three different aspects and try to develop an integrating approach and what kind of information we get in evaluating and understanding at these three different levels. One, at the mutation levels of EGFR, ER, PR, FOP2, BRCA, etc. Their mutations, their expressions. At the same time, the role of the microorganisms, their expression, their regulation. Apart from that, at the third level, the DNA repair genes, especially the mRNA. mRNA, what we call it as the checkpoint sensor genes. These three, if we try to integrate, would broaden our horizon of understanding the disease and its pathophysiology. Once we understand this, we try to develop the therapeutic strategies. So we are targeting and developing these platforms, integrative approaches, which would allow us to have a quick diagnostic approach. And based on this diagnostic approach, we can develop the therapeutic strategies. And we know that breast cancer is very, very complex. And that is the reason why we are trying to come up with these integrated approaches, which would help us identify at the diagnostic levels and specifically develop the strategies for the therapy. So again, at these three levels, we try to target with the drugs, different types of drugs, which in the succeeding slides, you are going to understand. The drugs targeting the expressed as well as the altered receptors of the biomarkers. Altered means the mutated ones. Then we have and developed an understanding of the microRNAs targeting the different biomarkers and their regulation. Once we have this understanding, then we thought of developing the therapeutic strategy by targeting these microRNAs with the drugs. And at the fourth level, we try to target even the DNA repair genes with specific drugs. So, if we carefully look at the EGFR receptors, we had tried to understand the amount of the mutation in EGFR, an unique one, at a specific position of the EGFR. And for this, we have screened almost more than 5,000 patients and try to understand the genetic alteration. The genetic alteration was at the 4%, which is considered to be huge. And within this genetic alteration, the missense mutation, specific mutation at a specific position, determines the high allele frequency compared to the other mutations. And we know that having the mutations coming up is a dynamic event. So we have to understand that among this dynamic event, which kind of mutation has the highest frequency, which determines the pathophysiology of the disease in that particular mutation. So when we have so many patients, we have come across that this mutation at this position is having a very high frequency. So we further looked into their overall survival rates as well as the relapse, and we see that the survival rates are very poor, and hence it becomes very important for us to target this risk. Further, we have carefully looked into this receptor, and as it was an mutation which has been identified, so we have used a lot of our understanding in developing this mutated structure which you see on the right. And if you see the normal one and the mutated one, at a specific position, you have this mutation. So when you have this mutation, you see that this biomarker has a different kind of the conformation which it tries to attain, because of which it becomes very difficult to challenge or target with the specific drugs. Conventional drug discovery tells us that if there is a change in the amino acid residue in the form of a mutation, and if that contributes to any kind of a structural change, then there would be the high probability that a specific drug which targets the normal EGFR might not be able to target the mutated one. But to our surprise, and which we have reported here, to our understanding, we have seen that though the structural changes or the conformational changes are happening in this receptor, a similar kind of a drug binds to both the normal or overexpressed EGFR as well as the mutated EGFR. And this is which belongs to the purine analogs. And we have almost screened 100,000 compounds of different functional groups belonging to the purine category. And the interesting aspect is that for the same compound out of 100,000, he is going to bind to both the normal as well as the mutated. So what does this tell us? It tells not only that a single compound, whether in case of cancer, if the aggressively EGFR is expressed and not mutated, which can be a contributing factor for the cancer, and if the same biomarker, if it is having a mutation at this specific position of 916, a single or the same compound can effectively target both the overexpressed EGFR as well as the mutated EGFR. Further, we have looked into to broaden our horizon of understanding about the interactions of this drug at both these levels, at the normal EGFR or the aggressively expressed EGFR and the mutated EGFR. And we see that it speaks volumes about the different types of interaction within the active site of both these EGFRs. And this is one area where we are further increasing our horizon in understanding and trying to get an even appropriate way of modifying this purine analog, which is the chemical synthesis process. And using some of the changes of the functional groups, we can effectively target even better both the normal as well as the mutated EGFR. So this is at one aspect of the integrative approach what we have used to develop. And similarly, we did a lot of studies to understand at different levels of their interactions. And similarly, we try to analyze the high frequency of the mutations which appear in the ER in the breast cancer patients. And for this, we have screened almost more than 700,000, 7,000 patients. And we have seen that even this mutation at this position of the ER receptor has very high frequency. And this is again another aspect which we have considered and try to understand. And we have seen that their interactions for both the normal as well as the mutated structures have a different kind of information which provides that both these areas of the normal, though highly expressed, as well as the mutated ones, try to have a different kind of interaction with the drugs with very high binding affinity. So, considering this as well as the EGFR, this clearly shows that how change in the receptor, change in the mutation, change in the structural information provides us information of what drugs would be efficiently binding so that we can develop a platform and use a specific strategy to treat to the patients. Then, as I mentioned, we have screened a lot of microRNAs, and we know that microRNAs basically are playing a key role in modulating the different aspects of the highly expressed receptors and their mutated ones. And we have screened a lot of microRNAs, and we have come up to a conclusion that microRNA 21 as well as 221 of more than 150 microRNAs, they are very specific to binding and regulating the ER receptors in the breast cancers. And once we have identified that, we are not sure what kind of structure they attain, because microRNAs are almost like 20 to 22 nucleotides long, and having the structural information is too difficult. But we have tried to understand the structure, and we have seen that the microRNA, these two microRNAs out of 150, these two have very high seed sequence complementarity. And then they have the GU pairing. And this GU pairing, because of the absence of GU pairing, they have very high thermodynamic stability. And the thermodynamic stability tells us that these structures, what we have understood, have a very good structural information that can efficiently bind to these receptors. Most of the time in the literature, we come across that the microRNAs in the experimental setups, they try to look whether they are present or not. But it cannot assure us that these microRNAs which are expressed can really bind to the specific microRNA, to the specific receptors, whether it be ER, EP, EGFR, PR, HER2, etc. So we have extensively looked into these aspects and tried to prepare a library of the complete set of microRNAs for each and every receptor, like EGFR, ER, PR, HER2, as well as BRCA1, BRCA2, and so on. So in all of them, we have observed that different microRNAs are having different kinds of structural stability, and they have different kind of a binding pattern to different kinds of the biomarkers or the receptors, which are expressed in the breast cancers. Similarly, for PR, we did the similar strategy, and we have come up with, again, different sets of compounds binding at different positions, both for the unaltered and the altered PR, etc. Similarly, we have looked into HER2, and we have obtained a different set of phytochemicals, series of compounds which bind effectively and differently to both the highly expressed as well as the mutated cultures. And in all these, we see that when it comes to the microRNAs, here we are only highlighting specifically microRNA 21 to 21 because of their efficient binding mechanism. Of course, we do have a list and a panel of microRNAs which have different binding approaches, different binding patterns to these different kinds of receptors, as well as to various kinds of drugs. And if we carefully look into these kind of phytochemical libraries, for all these receptors, ER, PR, HER2, as well as the BRCA1, BRCA2, we have almost screened 150,000 to 200,000 compounds of the phytochemicals. The main reason for considering this, as previously and aptly talked about by our previous speaker, more important understanding would be from the lead structure. So they have a backbone. Any drug, if we consider, they have a backbone, like this kind of a backbone or this kind of a backbone. And here we can have the different functional groups modified, and they determine the different binding affinities, interactions, efficiencies, as well as the kinetics with the receptors. And when we say about the phytochemicals, these phytochemicals tell us that they cannot elicit a greater amount of cytotoxicity as compared to the synthetic forms of the drugs. For this reason, we have chosen the library of the all mostly available phytochemicals, and we know that what out of the screen 150,000 to 200,000, we have again a panel of compounds, phytochemicals, which can bind efficiently, and of course these are all different phytochemicals, to be either altered, unaltered, or the same phytochemical can bind efficiently to both the altered and the unaltered cells. So what does this say? This says that these phytochemicals not only efficiently bind to the biomarkers, but may play a role in, again, the same compounds to either to the microRNAs or to the DNA checkpoint sensor genes. Again, mRNA is a complex which has the RAD50, MRE, as well as the NBN. Identifying this has given an understanding that what kind of drugs can efficiently bind to this. And after looking into this and a series of compounds of the phytochemicals, we have come up with a panel of compounds which can efficiently bind to all these DNA repair genes, which is MRE, mRNA, okay, mRNA, and this is the DNA repair complex having 3 different kinds of genes, and these are the different patterns. Then we try to analyze even further that how these mRNA genes or the checkpoint sensor genes are involved in interaction with the different kinds of the microRNAs. And we know that there are only a couple of microRNAs which bind to all the 3 DNA repair genes or the checkpoint sensor genes. So, and these 2 are nothing but our microRNA 21 and 221. And that is the reason in this presentation, I have only highlighted only 2 of them. And similarly, not only these 2, but these 2 are again interacting with your EGFR, ER, as well as the PR. So there are some microRNAs which can commonly bind to many kinds of receptor, not just one. So this is and again an interesting information what we have observed, that if we can and there is a way to screen and identify some compounds which can effectively bind to the microRNAs, to the DNA repair genes, as well as the different kinds of the receptors, whether it be EGFR, ER, PR, HER2, etc. We don't have to go with the combinatorial kind of a therapy or adjuvant therapy. A single compound can efficiently bind to all these markers which are having an integrating role. And that is the reason why we thought if we can develop an integrative approach and develop a platform which can suggest, after looking into all these markers, that which kind of drug, especially the phytochemical, having low toxicities and side effects, unlike the chemotherapeutic drugs which are more synthetic, can provide us a better way of the therapeutic approach. And similarly, if you can see here, we have tried to look and develop all these structures. And once after developing these structures of the microRNAs, then we try to interact with them with the specific mRNAs of either the mRNA DNA repair genes or the EGFRs or ERs, PRs, HER2s, BRCA, etc. So now we have so much of information for starting with the integrating approach and developing this platform. So we are in the process of and using AI and ML to have even more further understanding and develop a platform which can tell us that which kind of compound can be used effectively for which kind of markers which is highly expressed in the breast cancers. And of course, like I mentioned, we have looked into n number of panels of microRNAs, etc. And these are some of the other ones apart from 21, microRNA 21 to 21. And we have now a panel of the information. And after understanding further elaborated that if we are able to bind to these microRNAs or to these receptors, whether is there any deregulated role, because it is not just one pathway, there will be multiple pathways which might be affected. And we have tried to look into the disease ontology, reactomes, the KIT pathways, the wiki pathways, and try to come up with a solution that if we are able to target these specific microRNAs or the specific receptors or the DNA checkpoint sensor genes, whether they are going to affect the other pathways or not. And we have seen that the pathways which are related to the cancer, I mean, especially in the breast cancer, those all can be modulated as shown in this figure. And these can be very effective in way of the therapeutic approach. So in summary, I would like to say that we have identified various mutations, the new ones, and these would be very helpful in understanding each and every receptor's expression in the mutated form and the non-mutated form. And for each of these receptors, and again, a panel of drugs have been identified which can bind very effectively either the same drug or different drugs for the mutated and the non-mutated structures. Then we have reported that microRNAs regulate these receptor expressions differentially as compared to their normal counterparts. This is another understanding which provides even more understanding of the appropriate therapeutic approach. Then further, we have looked into the mutations in the DNA repair checkpoint sensor genes, which has differential roles in the breast cancer progression. And now we have a panel of drugs which can effectively bind either or all to the receptors microRNAs or the DNA repair genes at different levels. And their structural thermodynamic stabilities provide further evidence that these would be the best potential compounds, and our work in terms of both experimental as well as the clinical samples analysis are in progress, and we are getting some interesting results as per these studies, which we have already reported, and shows that this kind of an integrated approach will definitely help us not only from the effectively understanding at the diagnostic point of view, but similarly develop the appropriate therapeutic strategies. And these are some of the funding agencies, my collaborators, then my entire team here. I haven't put up with some of my alumnus here, but yes, they also directly or indirectly played a role in contributing to this extensive integrative platform development. And if you have any questions, quick questions, I can take a couple of them.
The uterus is a symbolic organ of womanhood. Hysterectomy is the second most common surgery done in women, next to cesarean section. According to WHO statistics, globally, about 1,540,000 women undergo hysterectomy in the year 2016. A study conducted in 2018 in India reported the prevalence of hysterectomy to be around 17 per thousand married women in India. Also, it varies among the states about 2 to 63 per thousand women. Hysterectomy is mostly seen among women aged 40 to 45 years, and by 65 years of age, approximately 37 to 39% of the women undergo this surgery. The most common social factors which contributes to the increased rates of hysterectomy is age of women, the age at marriage, literacy, and socioeconomic status. The need for the study. A review of several studies highlighted that the women experienced moderate to severe levels of anxiety and depression in the preoperative period, which continued even after surgery. And the psychological reactions associated with the hysterectomy is due to loss of uterus, limited physical and sexual activity, and fear of rejection by male partner. The preoperative teaching serves as a standard of nursing practice within the surgical setting. The evidence from several works of literature supports the preoperative instruction was effective in initiating early ambulation in the postoperative period to improve the pulmonary function and lessen the pulmonary complications. It also decreased the anxiety and depression and decreased the demand for additional analgesics and the total amount of analgesics. The setting was District Headquarter Hospital, which is the tertiary level hospital located in Salem, Tamil Nadu, India. The samples were the women who undergo hysterectomy and those who fulfilled the inclusion criteria. So based on the inclusion criteria, the experimental group was 184 women and control group were 95 women. And the sampling technique used were non-probability convenience sampling. The inclusion criteria, which includes the women between the age group of 30 to 60 years living with a partner, able to speak Tamil because the study was conducted in Tamil Nadu, those who undergo abdominal or vaginal hysterectomy for non-malignant reasons under general or spinal anesthesia. The exclusion criteria were women with debilitating illness, women with psychiatric illness, women who undergo hysterectomy for emergency reasons. The instruments used for data collection were proforma for the demographic data and the flow chart for vital parameters and other parameters, state and trait anxiety inventory, which is a standardized tool used to assess the anxiety, bed depression inventory tool to assess the depression, and checklist to assess the early postoperative complications. The preoperative teaching protocol for hysterectomy was given to experimental group, which included informations about general on surgery, the preoperative preparations and preparations on the day of surgery, the intraoperative care, and immediate and late postoperative care until discharge, including postoperative exercises, which covered respiratory exercises, mobilizing exercises, and muscle strengthening exercises. Following the teaching, the incentive spirometry was given to each subject in experimental group, and the researcher demonstrated to practice it in the postoperative period. The control group received routine hospital care. The ethical considerations. An ethical clearance certificate was obtained from the Ethical Committee of the District Headquarters Hospital, Salem, Tamil Nadu. Formal permission was obtained from the Dean to conduct the study in the gynecological wards of hospital. Informed written consent was obtained from each subject. Further, the subjects were assured of privacy and confidentiality of the data collected. This table describes how the data collection was done in different periods. So in pre-test was conducted in the preoperative period, which include using the instruments of demographic data, physiological vital parameters, which includes vital signs and inspiratory capacity, and other parameters which consist of the length of stay, the amount of analgesics consumed, parenteral analgesics administered, and the demand for additional analgesia, including early ambulation. And anxiety and depression, these was measured in the pre-test, again in the post-test file at the time of discharge. The early postoperative complications were monitored throughout the stay in the hospital in the postoperative period. So the data analysis, the collected data was analyzed with the SPSS software, and descriptive and inferential statistics were applied to make the interpretation of the study findings. So the table 1, which describes the distribution of subjects based on the demographic data. In experimental group, majority of women belongs to 36 to 40 years, whereas in control group, majority were in 41 to 45 years. And educational status in both the groups, majority of them were illiterate, and occupation wise, most of them were coolie workers. The family monthly income, which was less than 2,000. This study was done in the year of 2015 to 16. So that time, so it's coded as less than 2,000. So majority had that much income. And the number of children, most of them had 2 children. And the duration of compliance were less than 3 months in both the groups. And regarding the preoperative diagnosis, this diagram, which clearly explains majority women were suffering from fibroid uterus in both the groups, followed by dysfunctional uterine bleeding was the major complaint, and genital prolapse and also adenomyosis. And only 1 woman suffered with chronic cervicitis in experimental group. Regarding the type of surgery undergone, majority of the women underwent total abdominal hysterectomy along with bilateral salpingo-oophorectomy in both the groups. And few of them have underwent total abdominal hysterectomy. And as some of them suffered with vaginal prolapse, uterine prolapse, so they have undergone vaginal hysterectomy along with the pelvic floor repair. And few of them also had vaginal hysterectomy. Regarding the natural menopause, which was attained by the women before the surgery, majority were having regular menstrual cycle. So this is a very important finding in the study. And further, the duration, majority had even though they had a natural menopause, it stopped within 1 year. And associated urinary symptoms were none of the majority didn't have any urinary incontinence in terms of stress, urge, or mixed incontinence. Comorbid illness. Majority did not have comorbid illness in both the groups. Coming to the assessment of physiological vital parameters, which mainly consist of vital signs, which were found to be normal in terms of temperature, pulse, and the respiration in the experimental group, and also systolic blood pressure and diastolic blood pressure, and significantly the inspiratory capacity, which was normal in the preoperative period, about 47.3% of women had a normal inspiratory capacity of 1200 ml, but during the early postoperative period, that is within 12 hours and within 24 hours and on the second postoperative day, it started to reduce, but again, it started to regain on the 5th postoperative day and at the time of discharge in the experimental group. This is the findings of control group, where majority maintained normal vital signs of temperature, pulse, and the respiration, systolic blood pressure and diastolic blood pressure. However, the very significant numerics to be noted here is the inspiratory capacity, in which majority had normal in the preoperative period. However, at the time of discharge, only 19% of women had the normal inspiratory capacity, which was 1200 ml. Further, the statistical test, which describes the difference between the experimental and the control group, revealed the inspiratory capacity had a very significant findings between the experimental group who used the incentive spirometry as compared to the control group. The ambulation, the experimental group majority ambulated within 24 hours after surgery, whereas in control group, only 48, after 48 hours, majority have ambulated. And regarding demand for additional analgesics, it was majority did not take the, didn't request for an additional analgesics in both the groups, and the total amount of parenteral analgesics received were within 3 to 4 doses. And the length of stay, again, in the experimental group, the mean length of stay was around 91% of women stayed longer as compared to control group, where majority of them stayed within 11 to 15 days. Further, the statistical analysis clearly describes here the experimental group, the mean ambulation hours was 30.33 as compared to the control group, which was 41.56, which had a statistical significance. Further, the length of stay, the mean length of stay in the experimental group was 17.65 days as compared to 14.31 in control group. Further, on the anxiety assessment, on pre-test as compared to pre-test and the post-test, between the experimental and the control group had a significant reduction of anxiety on the group which received the preoperative teaching protocol. Again, these findings were statistically significant. Same way, the depression also at pre-test, as compared to pre-test, the post-test, there was a significant reduction of depression scores in the experimental group as compared to the control group. And in regard to the early postoperative complication rates in the experimental and control group revealed, the most significant complications were bleeding, pulmonary complications was nil, constipation, which was equally seen in both the groups, but very significantly, the wound infection was more on control group as compared to the experimental group. The discussion. The measurement on physiological vital parameters revealed majority had normal vital signs. There was no significant difference found between the subjects except for inspiratory capacity. The effective practice of deep breathing, controlled coughing exercises, and use of incentive spirometer in the postoperative period helped the experimental group to achieve positive results. Above findings were consistent with other studies taken by the different authors. The experimental group ambulated earlier as compared to control group, which had a statistical significance. And most of the subjects in both the groups did not demand for additional analgesic and received only 3 to 4 doses in the postoperative period. The length of stay was more in experimental group, which was average of 17 days as compared to control group, where 14 days. So the reason for this is in government setting, it was noted that there was lack of policy on the total length of stay after gynecological surgery. Many times it was left to the women's preferences. So most of them opt to stay in the hospital to take more rest than in the home settings. So these factors contributed to the increase in the length of stay for the present study. Similar findings also were noted by other authors as well. The pre-test mean anxiety and depression score was the same in the experimental group and control group. But after administration of the perioperative teaching protocol, the mean anxiety and the mean depression score was lower in the experimental group during post-test 1, which had a statistical significance. And the early post-operative complications rate was higher in the control group as compared to the experimental group. So the implications of the study: the nurses in the gynecological ward can provide structured perioperative information about surgery and its preparation, including the postoperative care, which will reduce the anxiety and depression in the women and facilitate early participation in self-care postoperative activities. Teaching and demonstration of postoperative exercises will aid in early ambulation and reduce the incidence of postoperative complications. The nurses can prepare women about home care and management of after effect symptoms due to related to surgical menopause. Reproductive tract problems have to be dealt with alternative approach to avoid early hysterectomy done at the younger age. These were the references used for this presentation. Thank you.
Okay, thank you very much, Dr. Tilakavati.
Srinagar, Jammu and Kashmir, India. So my topic is induction versus adjuvant chemotherapy combined with concurrent chemoradiation. What is beneficial in locally advanced nasopharyngeal carcinoma? And this is a 5-year comparative study at a tertiary care center in North India. Nasopharyngeal carcinoma is an epithelial malignancy arising from the mucosa of the nasopharynx, most of which occurs in the upper and lateral walls, particularly in the pharyngeal recess. Nasopharyngeal carcinomas constitute 60% of all head and neck cancers, and it is one of the most frequently diagnosed head and neck cancer. As per the International Agency for Research on Cancer, 130,000 new cases of nasopharyngeal cancer has been estimated in the year 2018 worldwide, and it is accounting for 0.7% of all cancers diagnosed in 2018 worldwide. The incidence is very high in the Southeastern Asia, Southern China, and Northern Africa. Coming to the incidence to our part of the world, it's very, very less as compared to the other part of the world. As per our population-based cancer registry, the incidence was 0.82 in males and 0.24 in females in 2020. Nasopharyngeal cancers, they are very notorious. They cannot be detected at an early stage because of their deep and hidden location. That is why more than 70% of the patients have advanced disease at presentation. Because of their radiosensitive nature, radiation therapy plays an important role in the management of early stage nasopharyngeal cancer, and we can achieve 87 to 96% of 5-year overall survival with radiation therapy alone in early stage nasopharyngeal cancer. However, in locally advanced nasopharyngeal cancer, the combined modality approach is the standard of care. Intergroup 0099 was the first clinical trial to establish the concurrent chemoradiation followed by adjuvant chemotherapy in locally advanced nasopharyngeal cancer, both in terms of the progression-free survival as well as the overall survival. But unfortunately, majority of the patients, they cannot complete the adjuvant chemotherapy because of severe toxicity of concurrent chemoradiation. And also a number of the RCTs have shown no added benefit of adding adjuvant chemotherapy followed by concurrent chemoradiation as compared to the concurrent chemoradiation alone as far as the survival is concerned. So all these things limits the widespread use of adjuvant chemotherapy. Here comes the role of induction chemotherapy. So induction chemotherapy is an option, and it attracted lots of attention with better patient compliance, and also eradicates the micrometastasis. And also a number of phase 3 randomized controlled trials proved a survival advantage of induction chemotherapy followed by concurrent chemoradiation as compared to the concurrent chemoradiation alone. So the aims and objectives of this study is to compare the efficacy of induction chemotherapy and adjuvant chemotherapy combined with concurrent chemoradiation in locally advanced nasopharyngeal cancer. This study was a retrospective study which was conducted at Sher-e-Kashmir Institute of Medical Sciences, Srinagar. The patients' data were collected from October 2016 to June 2022. The primary endpoint of the study was overall survival, and the secondary endpoints were the loco-regional relapse-free survival, failure-free survival, and distant metastasis-free survival. We have taken a total of 80 patients in our study. Although this was a retrospective study, so we have not calculated the sample size prior. So we include all the eligible patients during this study. So we have included patients of nasopharyngeal carcinoma with stage 2 to 4A as per the 8th edition AJCC staging system. Patients who had received induction chemotherapy with concurrent chemoradiation, patients received adjuvant chemotherapy with concurrent chemoradiation, and we have excluded patients with stage 4 disease and those patients who had received only concurrent chemoradiation. Routine pre-treatment evaluation has been done in all the patients, including complete history and physical examination, routine blood test and biochemistry profile, mirror and fiber optic examination, magnetic resonance imaging of the nasopharynx and neck, computed tomography of the chest with or without contrast, and whole body bone scan or PET scan if indicated. All patients received radiation therapy with intensity modulated radiotherapy. The target volume delineation was done according to the International Commission on Radiation Units and Measurement Report 50 and 62. The planning target volume of the primary tumor received 70 to 72 grays. The PTV of the high-risk regions received 60 to 63 grays, and the PTV of the low-risk regions received 50 to 54 grays. All patients received one fraction per day, 5 days a week. Coming to the chemotherapy part, in the induction chemotherapy, we have cis regimens used were cisplatin and 5-FU. In the cisplatin and 5-FU regimens, patients received injection cisplatin 100 mg per meter square on day 1, injection 5-FU 1000 mg per meter square, continuous infusion for 4 days. Then we have another cisplatin and docetaxel regimen. Patients received docetaxel 75 mg per meter square day 1 and injection cisplatin 75 mg per meter square. And another regime was the cisplatin 5-FU and docetaxel, where patients received docetaxel 70 mg per meter square on day 1, cisplatin 75 mg per meter square on day 1, and injection 5-FU 1000 mg per meter square, continuous infusion for 4 days. All patients received 2 to 3 cycles of induction chemotherapy, and each cycle was repeated after 21 days. In the adjuvant setting, patients received mainly the cisplatin and 5-FU regimens, and some of the patients also received single-agent cisplatin. And again, the cycles were repeated every 3 weekly, and patients received 3 to 4 cycles of adjuvant chemotherapy. In the concurrent setting, patients received either injection cisplatin 40 mg per meter square weekly or 100 mg per meter square 3 weekly with radiation therapy. Coming to the results, the baseline characteristics were well balanced between the two groups. The median follow-up was 50 months in the induction chemotherapy group and 55 months in the adjuvant chemotherapy group. However, the survival was different between the two groups. Coming to the demographic profile, as you can see, In the induction chemotherapy group, 37.5% of the patients were aged more than 51 years. 25% of the patients were in the age group of 37 to 41, and 20% of the patients were in the age group of less than 36. Whereas in the same as in the adjuvant group also, 45% of the patients were more than 51 years, followed by 22.5% of the patients were in the age group of 37 to 41, and 17.5% is in the age group of less than 36. In the induction chemotherapy group, 92.5% were males, while only 7.5% were females. While same in the like in the adjuvant group, 87.5% were males and only 12.5% were females. In the induction chemotherapy group, 70% were smokers, while 30% were non-smokers. In the adjuvant chemotherapy, 65% of the patients were smokers, while 35% were non-smokers. Coming to the histopathology, in the induction chemotherapy group, 72.5% of the patient had WHO type 3 histology, and 20% had WHO type 2 histology, and only 7.5% have WHO type 1 histology. Similarly, in the adjuvant group, 70% of the patient had WHO type 3 histology, followed by 27.5% had type 2, and only 2.5% had WHO type 1 histology. In the induction chemotherapy group, 47.5% of had T3 stage, and 25% had a T4 stage, and 15% of the patient had a T2 stage. Similarly, in the adjuvant chemotherapy group, again, 45% of the patient had T3 disease, 22.5% of the patients had T4 disease, and 17.5% of the patient had T2 disease. Coming to the end stage in the induction chemotherapy, 52.5% of the patient had N1 disease, 20% of the patient had N2 disease, and 15% of the patient had N3 disease. Similarly, in the adjuvant group also, majority of the patient, 50% of the patient had N1 disease, 17.5% of the patient had N2 disease, and 12.5% had N3 disease. Coming to the chemotherapy regimens, in the induction chemotherapy, group, 40% of the patients received cisplatin and 5-FU regimen, followed by 30% of the patients received cisplatin, docetaxel, and 5-FU regimen, followed by 20% of the patients received docetaxel and cisplatin regimen. Whereas in the adjuvant chemotherapy group, 60% of the patients received cisplatin and 5-FU based chemotherapy, and 32.5% of the patients had received only single agent cisplatin chemotherapy. Coming to the number of the cycles, in the induction chemotherapy group, 60% of the patients received 2 cycles of chemotherapy, while as 32.5% received more than 2 cycles of chemotherapy, while as in the adjuvant group, 50% of the patients received more than 3 cycles of adjuvant chemotherapy, followed by 35% of the patients received 2 cycles of adjuvant chemotherapy. Follow-up duration was measured from the day of the initial pathological diagnosis to the last visit or death. So these patients were followed every 3 monthly during the first 2 years, then every 6 monthly thereafter. We have calculated the overall survival from the date of treatment to last date known, alive or death. Failure-free survival was calculated from the date of treatment to the failure, death, or last visit. Local regional relapse-free survival was calculated from the date of treatment to the local or regional relapse, and distant metastasis-free survival was calculated from the date of treatment to the distant metastasis. Here, this slide is showing the survival in both the groups. Here you can see the induction chemotherapy group had a better survival as compared to the adjuvant chemotherapy group. 90% versus 81%. The failure-free survival is 80% versus 77.5% respectively in the induction chemotherapy group and adjuvant chemotherapy group. Distant metastasis-free survival is better, 88% versus 82.5% in the induction chemotherapy group. Only the loco-regional relapse-free survival is better in the adjuvant chemotherapy group, that is 92% versus 90%. But this was not statistically significant. On subgroup analysis, we have found a statistically significant difference in the overall survival in the induction chemotherapy group, that is 94% versus 89%, with a p-value of 0.012. Failure-free survival 90% versus 89% with a p-value of 0.039. This loco-regional relapse-free survival 95% versus 89% with a p-value of 0.035, except the distant metastasis-free survival, which was same in both the groups. So additional chemotherapy with concurrent chemoradiation is promising treatment option for locally advanced nasopharyngeal cancer. However, the optimal timing of additional chemotherapy remains unclear. Number of the systematic reviews suggested that induction chemotherapy prior to concurrent chemoradiation is associated with superior overall survival and progression-free survival. As per the ASCO 2021 consensus, it recommends induction chemotherapy followed by concurrent chemoradiation in loco-regionally advanced stage of nasopharyngeal cancer. Currently, the only phase 3 randomized controlled trial, which is evaluating the survival difference between induction chemotherapy and adjuvant chemotherapy combined with concurrent chemoradiation, is the NCT 03306121 trial, the results of which is awaited. Our study revealed induction chemotherapy with concurrent chemoradiation provides a favorable overall survival. A subgroup analysis based on the T-stage category was done and showed that induction chemotherapy concurrent with chemoradiation improved the overall survival, failure-free survival, and loco-regional relapse-free survival in T3 nasopharyngeal carcinoma, while as adjuvant chemotherapy with concurrent chemoradiation improved the loco-regional relapse-free survival in T4 disease. On stratified analysis revealed a significant 5-year overall survival in patients with N2 nodal disease treated with induction chemotherapy followed by concurrent chemoradiation. There are certain limitations of our study. One is the retrospective nature of the study. And because of this retrospective nature of the study, we could not analyze the acute toxicities, and we were not able to compare the acute toxicities between the two groups. And a second is there is uneven distribution of the chemotherapy regimes between the two groups. And third is in some of the patients in the adjuvant setting, patients received only single agent cisplatin because of toxicity of the 5-FU. So with this, we revealed survival benefits of induction chemotherapy followed by concurrent chemoradiation in patients with stage T3 and N2 nasopharyngeal carcinoma, while as adjuvant chemotherapy with concurrent chemoradiation improved the loco-regional relapse-free survival in T4 nasopharyngeal cancer without any other survival advantage. In the future, further research is needed to prove whether adding chemotherapy before rather than after concurrent chemoradiation should be recommended in locally advanced nasopharyngeal carcinoma, especially considering the possibility of adverse reaction. Thank you all for your attention.
Thank you very much, Dr. Canis. It was a wonderful presentation. It was very interesting. So I don't know
my name is Dr. Hind Hari. I am from the Faculty of Medicine, International University of Africa, and I am happy to be here at the international webinar on breast cancer and therapeutics. The outline of my presentation will be the objectives, literature review, the background, of course, at the first, methodology, pathophysiology, results, and the conclusions. Preeclampsia is a diverse multi-organ group of related disease processes and it is a disease of second half of pregnancy, starting from 20 weeks of gestation, and it is usually causes 5 to 7% of all pregnancies, and this may rise in developing countries like Sudan. And it is also known by having a high effect on maternal and fetal morbidity and mortality. The objectives of this study was to assess changes in complete blood counts, including platelets, liver enzymes, and serum uric acid in preeclamptic cases, compared to second half normal pregnant and non-pregnant Sudanese women and their correlations to other biomarkers. Our literature review in hypertensive disorders in pregnancy is that they cause 20% of maternal death, and usually they constitute 30% is from chronic hypertension, and the 70% are caused by gestational hypertension. The hypertensive disorders in pregnancy can be either chronic hypertension or pregnancy-induced hypertension. The pregnancy-induced can be gestational hypertension without protein in the urine, or preeclampsia with protein in the urine. And this preeclampsia can be either mild or severe, and it may go to impending eclampsia, which is associated with convulsions of the patient. The chronic hypertension can lead to chronic superimposed by preeclampsia. So it is a disease of the second half of pregnancy, starting from 20 weeks of gestation. It can be either starting before pregnancy, and this is chronic hypertension, and it can lead to, or it is preeclamptic or gestational hypertension, which appears at the week of 20 of gestation and onwards. So it is a multi-system disorders affecting 5 to 7% of all pregnancies. The predisposing factors can be placental, genetic, immunologic, and environmental, which may interact. The methodology, this was a cross-sectional case control study performed in Omdurman Maternity Hospital. The sample size included 3 groups, 72 preeclamptic cases, 96 normal pregnant in their second half of pregnancy, and 63 non-pregnant as control women, a total of 231 subjects. Preeclampsia can be either mild, in which the blood pressure ranges in or above 140, the systolic and the diastolic about 90 or more than 90. And this is the mild, with proteinuria 1 dipstick, 1+ dipstick test, or 300 milligrams per 24 urine sample urine. It can be severe, and the blood pressure is higher, 160 milligram mercury, the systolic and the diastolic more or equal to 110 milligram, and the dipstick proteinuria is 3+ or 4+. As I mentioned, it is a disease of second half of pregnancy, and major changes occur in the placental level. This is causing ischemic placenta. The causes can be increased AT1 angiotensin 2 type 1 agonistic autoantibody, or nuclear cells, or genetic factors, or oxidative stress. These all affect, causing ischemia in the placenta. And the ischemic placenta either affects the mother directly, or as well the fetus, causing growth restriction of the fetus. In the placental ischemia, it causes changes in the maternal organs, the major organs affected, the kidneys, the liver, and the brain. and the endothelial cells lining of the organs. The severe, as I said, it can be severe and this leading to convulsions, or it is characterized by a pitting edema, the urine proteinuria, and the buffy face with visual disturbances, and of course, increase in the body weight of the mother. This figure shows the changes which occur at the level of the kidney. There occurs endothelial cell, swollen, non-fenestrated endothelium, subendothelial fibroid deposition, and mesangial cell interposition. So systemic hypertension can lead to effect over the kidney in the glomerular kidneys, glomeruli of the kidneys, and this in the vas efferents, and this causes hyperperfusion, and this can lead to decreased glomerular filtration rate or affecting directly the endothelial cell lesions, and this as well it decreases the coefficient of glomerular filtration rate. The analysis was done at St. Heliers Hospital in London, the biochemical analysis of this data. Our results were that the mean hemoglobin concentration of the preeclamptic was statistically significantly lower than that of the non-pregnant, but not from that of the normal pregnant. There was no statistical significant difference in the mean white blood cell count between the preeclamptic and non-pregnant, and between the preeclamptic and the normal pregnant. There was a considerable statistically significant decrease in the mean platelets count of the preeclamptic compared to the non-pregnant, as well as to compare to the normal pregnant. In the preeclamptic cases, the ALT, alanine transferase, correlated significantly with the total white cell count and with the serum aspartate transferase. In the preeclamptic cases, serum AST correlated significantly with the hemoglobin, with the serum ALT and serum uric acid. There was a significant and weak negative correlation between albumin and systolic blood pressure. The higher the albumin, the lower is the systolic blood pressure. The study population was 231, the preeclamptic 72, the normal pregnant 96, and the non-pregnant 63. The age group ranges from 19 to 45. Here, the gestational age of the pregnant subjects. And here it shows the serum creatinine levels, the serum cystatin C, serum uric acid, and the glomerular filtration rate. And as you can see, the GFR, or glomerular filtration rate, is dropping in the preeclamptic cases compared to the non-pregnant. This diagram as well shows the differences between the serum levels and the GFR of the three groups of subjects. Here, serum albumin, serum aspartate, and the serum alanine aminotransferase levels. As you can see, is a significant difference between the non-pregnant and the preeclamptic levels in the serum albumin. Also here, it shows that there is a significant difference between the normal pregnant and the preeclamptic in the serum aspartate level. Here shows the complete blood count. We can see the significant changes between the non-pregnant and the preeclamptic in the hemoglobin, in the platelet count. It dropped from the non-pregnant to the preeclamptic. But the white count, there is no significant changes. And between the normal pregnant and the preeclamptic, there was also significant drop between the normal pregnant and the preeclamptic. Here, this histogram of platelet count shows the differences in levels for the platelet count for the three groups, starting from less than 100,000 platelets per cubic millimeter up to 499 level. And as you can see, most of the cases of our cases for the preeclamptic, it lies between 200 and 299. And also most of the cases between 100 and 199 were the preeclamptic cases. So there is significant drop in the platelet count of the preeclamptic other than the two groups. This also shows the hemoglobin level changes between the three groups. Here also we see the correlations and the pre-values. You can do the readings, systolic blood pressure, diastolic blood pressure, vertical column with the horizontal column, and you can see the level of significance and correlation for each of our variables in this table. So our conclusion, there was a considerable statistically significant decrease in mean platelet count of the preeclamptic compared to the non-pregnant group and to the normal pregnant. And this can be explained by the hemodilution. As we know, in pregnancy, hemodilution occurs. But further drop between the normal pregnant and the preeclamptic platelet count can be explained by the pathology which occurs due to preeclampsia, and that may be related to increased coagulation and which can lead to acceleration of consumption of the platelets count in the preeclamptic cases. And these are my references, and thank you very much.
Thank you very much, Dr. Hind. That was a lovely presentation. Let me see if there are some questions. Okay. First, I'm going to thank.
Side effects. It's a case report, encephalopathy and status epileptics. I will talk in this presentation, introduction for 5-FU chemotherapy, side effects and rare side effects for 5-FU. And also I will explain the cases that have been presented to our critical care units, the patient's information and the clinical finding when the patients admitted to our units, and the therapeutic intervention that have been done for the patients during his staying in ICU, and follow-up and outcome for the patients. And also I will explain the treatment modalities that have been suggested in literature regarding treating 5-FU encephalopathy, and conclusion of my presentation. So as we know, 5-FU is anti-metabolite and anti-neoplastics agents that have been widely used as adjuvant chemotherapy for variety cancer, such as carcinoma of the breast, pancreas, and colon, and rectum, and the stomachs. And also continuous infusion of 5-FU is a first-line therapy for metastasis breast cancers. Additionally, 5-FU infusion might be used in chemotherapy treatment regimen for invasive breast cancers, include triple-negative breast cancer. The side effects.
Sorry to interrupt you, but we cannot see your presentation. Can you share the screen with us, please?
Yeah, it's not shared.
No.
As well as myelosuppression and hand and foot syndrome. Additionally, chronic coronary vasospasm. About 5% of patients who receive 5-FU have been reported to experience neurotoxicity symptom, which described hyperammonia encephalopathy, Werin's encephalopathy, cerebral ties syndrome, and subtype multifocal leukoencephalopathy. There is patients reported in our hospital admitted to critical care unit after he took first session of 5-FU chemotherapy. This patient's 51 years old male patients with history of hypertension, diabetes mellitus, and chronic kidney disease, and estimated glomerulus filtration rate 12.5. The patients underwent EGD related to epigastric pain for 1 year. The EGD results that there is a mucosal lesion 10 centimeter by 5 centimeter. The biopsy result shows that patients gastric adenocarcinoma, have gastric adenocarcinoma with no evidence of metastasis disease. The patients underwent subtotal gastrectomy. The histopathology of biopsy shows that T and M staging for these patients, T it was 1, N it was 1, metastasis it was 0. The patients underwent chemotherapy and commenced in single agents 5-FU in adjuvant setting related to low glomerulus filtration rate and high comorbidity. The chemotherapy dosing for these patients was followed based in body surface area. The body surface area for these patients, it was 1.74. Firstly, the patients take calcium gluconate, folate, 400 milligram IV, then a bolus 5-FU chemotherapy 400 milligram. Then he took infusion 5-FU 2,400 milligram after continuous infusion for 46 hours. After 6 hours of completion, the first dose, the patients developed altermentation and seizure. Then the family of the patients brought him to our hospital. Upon admission, the patient was hemodynamically stable. GCS, it was ranging from 8 to 10. He was drowsy with equal and symmetrical pupil size. He was actively seizing with intermittent right side neck turning and tonic-clonic seizure, lasting for a few seconds to minutes, with no regaining of consciousness in between. Lab tests have been done for that patient. It shows that creatinine, it was elevated. It was elevated more than before. And C-reactive protein also, it was elevated. The results of complete blood count, serum electrolyte, glucose and ammonia and thiamine, and blood pH and liver enzymes was normal. Chest X-ray and brain CT scan without contrast have been done for that patient, but it doesn't show any significant results. Lumbar puncture have done for that patient. The result shows that leukocytes, it was 2 in CSF. CSF glucose was 6.2 and CSF protein level was 244. CSF gram stain and culture were negative, and acid-fast bacilli and fungal smears along with CSF viral screening were negative. Serum glucose level, it was 8 millimole. CSF paraneoplastic antineural antibodies were negative. CSF autoimmune encephalopathy antibodies, serum paraplastic antibodies, neural antibodies, and serum autoimmune encephalitis antibodies was negative. The DBU genotype was intact. MRI and EEG have been done for that patient. The MRI show hyperintense signal involving bilateral thalami along with bilateral cortical of occipital lobe and hippocampus. This is the results or the primary results of MRI for our patients. As you see here, there is hyperintensity signal in hippocampus and thalami for the patients. EEG records showing that there is epileptic discharge arising from temporo-occipital lesion. The patients, when he admitted to our unit, GCS, it was ranging from 8 to 10. For this cause, the patients intubated and refractory status epilepticus was managed. Anti-epileptic medication include levocitran, valproate, lacosamide, and tobromate, along with IV thiamine was started for him. Later, IV methylprednisolone was given for 7 days, along with course of treatment. The patient's electroseizure was controlled, and EEG showed diffuse slowing background activity. 2-week post-admission in the ICU, the patient shows mild clinical improvement in terms of his consciousness level. He extubated and remained seizure-free. After 9 weeks of initial presentation, IV methylprednisolone 1 milligram and IVIG given for 5 days for the patients. After that, the patient showed improvement in his consciousness level and his comprehension. His disorientation improved over time during the hospitalization stay. The patient was physically rehabilitated and discharged home. 2 months followed up in outpatient setting. He was functionally independent, alert, oriented, and minimal memory impairment. Brain MRI shows partially improvement in hyperintensity in bilateral thalami, but persistent hyperintensity signal in occupation cortex. This is the results of MRI after 2 months. And here we can see that there is improvement in hyperintensity signal, whereas the region it was present before. There is many treatment modalities have been presented in literature regarding 5-FU encephalopathy. It's ranging from suppurative measures to use of corticosteroids, plasma exchange, and thiamine, along or in combination, shows therapeutic consistency. The mainstay of treatment 5-FU encephalopathy antidote for 5-FU. Uridine tricyclase have been suggested in the literature and should be considered in severe cases of dehydropyramide dehydrogenase deficiency. Although the exact mechanism that underlie the various neurotoxicity are poorly understood, several theories has been proposed, include Krebs cycle suppression, that have suggested the cause of encephalopathy is related to fluorocytase, which inhibits the adenosine triphosphatase dependent urea cycles, causing hyperammonia and lactate acidosis. The conclusion of my presentation, as we know, 5-FU penetrate blood-brain barrier and can result in neurotoxicity, which is uncommon side effect with a described in literature with incidence rate 5%. It could be acute or delayed reaction. Acute neurotoxicity usually presents with hyperammonia encephalopathy, Werin's encephalopathy, and cerebral type syndrome, and delay in neurotoxicity manifests as subacute multifocal leukoencephalopathy. The diagnosis base for 5-FU induced encephalopathy include the development of encephalopathy shortly after 5-FU administration, along with exclusion of other medication, somatic, drug-related causes. It's described in literature that DBD deficiency. DBD is an enzyme responsible for a breakdown of 5-FU, and its deficiency leads to 5-FU accumulation with a neurotoxic effect such as demyelination and increased cellular thiamine metabolism, leading to Werin's encephalopathy-like manifestation. The different side effects might be having, such as stroke, encephalopathy, central nervous system infection, and psychogenic disorder. 5-FU related encephalopathy is rare, but potential side effect. The patients can present with different stage of alter of sensorium. The presence of hyperammonia can lead to diagnosis, but normal level of serum ammonia does not exclude the diagnosis. It's crucial for oncologists and the neurologists to remember and understand this morbid side effect, so the promoted treatment and diagnosis can be made with 5-FU, and supportive treatment is ensured. Thank you for listening, and I am happy to receive any questions.
Thank you very much for your presentation. Okay, let me see if radiologists are the key to a good diagnostic now, and everything will start with that diagnostic. So we need our radiologists to be happy now, to be comfortable with their work. So at first, I want to have the disclosure that I'm not a psychologist. So first, I'm going to introduce you to burnout syndrome. It was first defined by Frendenberg in 1975. He was a psychologist, and he was working in clinic with drugs problems. So he was noticing that the people that they were working in that clinic, they changed the behavior, no? So afterwards, in 1976, Christine Maslach, that she still lives in United States, she has 77 years old, and Jackson defined the burnout like an overload situation between professionals and developed the idea that it's constituted by three components, items or dimensions: emotional exhaustion, depersonalization, lack of personal or professional fulfillment. So at the end, Maslach and his collaborators created the most used questionnaire, Maslach Burnout Inventory Services Survey, that is used whole wide world to manage the burnout syndrome. So the definition by the 11th International Classification of Disease Stress in the Workplace that has not been managed successfully. It is characterized by three dimensions: feeling or of lack of energy and exhaustion, increased mental distance from work or negative feelings or cynical attitude towards work, a sense of ineffectiveness and lack of realizations. So if we go back to the Frankenberg clinic, we can imagine that burnout syndrome starts with emotional exhaustion. Imagine that people that's working all day with drug problems, at the end they feel exhausted because every day they have the same problem now, because it's difficult to treat with drug problems. At the end starts the second part, the depersonalization, where I'm very cold with that story, so it doesn't matter for me. And afterwards, the third step is where I feel that I'm not good at my job and I have problems with myself. Okay, so now we're going to start to take every part. The emotional exhaustion. It is related to an individual's experience of chronic stress that produces a progressive loss of energy that in turn is related to a decrease in emotional and physical resources. The sign is the lack of energy, impatience, irritability, suspicious thoughts. The depersonalization on the scene is, the term implies disconnecting from work as a response to the overwhelming amount of demands placed on it. The loss of enthusiasm and passion for work activity is linked to a negative attitude that manifests itself as indifferent and negative. Signs: loss of career expectation, attitudes of distancing and coldness, loss of empathy. And the last, the professional achievement or self-fulfillment, it refers to a decrease in professional efficiency and labor productivity. Signs: negative attitude towards oneself, gradual decline in self-esteem, deterioration of work performance. So burnout in medicine, because I think that all this syndrome is not just for the radiologists, but all doctors in general. And it has very important consequences. They can be, we can talk about the professional consequence, that normally the professionalism is being eroded, the quality of healthcare is negatively impacted, and we have increased the risk of medical errors. And personal consequences normally appears relationship breakups, emerge of suicidal thoughts. I think we know that doctors generally, we have a lot of suicide cases. Problematic consumption of alcohol and illicit substances or a lot of drugs. and the rise the risk of cardiovascular events, metabolic syndrome, or impaired immunity. Now, yeah, I think all of us know a doctor that with 50 years old, it has diabetes, a heart attack, and so on. So this is some studies. You can see that they are made in France, Switzerland, and Denmark. And we see that there are like a very high percentage of burnout. If we compare Denmark with Switzerland, for example, now we see Denmark is 25%, and we say, okay, oh, it's not so much compared to 70% of Switzerland. But you can see that 25%, it means one from each 4 doctors. So it's a lot. And these meta-analyses that were made all over the world, they are similar to the previous results. Talking a little bit of burnout syndrome in radiology, it's very unhyped in the last years, because before nobody spoke about that. So according to a national survey conducted in the USA, 79% of radiologists are affected by this syndrome. And in a Medscape survey in 2013, radiology was the 18th specialty in the category of burnout in medicine. So almost 36% of radiologists. And in 2020, they were placed in the 6th position with 46%. Now that was a very huge increase of burnout in very few years. I think that the explanation is a lot of the images that now we have to read, and every day they are increasing with a level that we cannot keep up. No, so I wanted to talk about the novel coronavirus pandemic, because I think it's important too. Everyone knows that on the 11th of the March 2020, it was declared the pandemic. And it was seen that in the first months, a significant increase of emotional exhaustion and depersonalization was seen among all healthcare workers, but especially for the radiologists, because in the COVID-19 disease, it had typical patterns of pneumonia with a lot of systemic complication. And we had to have a very high speed adaptation for that new disease. So now I'm going to talk about the burnout in Spanish radiologists, because this was my PhD. It was in the Hospital La Fe de Valencia with Dr. Luis Martín Bonmatí. So when I was doing my my PhD, the pandemic happened. So at the end, it was divided, my PhD, in 3 different time of periods. The first 2, it was with just a Google Forms application with a MASLAC questionnaire that I showed you before. The first period, it was January to July 2019. The second period, it was 5 months to April to August 2020. And the last one, it was just in the hospital. So the prevalence of burnout in the Spanish radiologists before the pandemic, it was 1 of 3 radiologists, 33.6%. And during the pandemic, it was 1 of 2 radiologists, 49, almost 49%, or almost a half. No, it was a very rapidly increase of burnout. So after we studied a lot of variables, I would like to say that no statistically significant correlation between burnout and social demographic variables of respondents were seen, like age, gender, autonomous community, marital status, number of children. I think that's important because in the literature, we can see that it was associated, but nowhere it could be found. So that shows us that the burnout syndrome is very more complex. And talking about the professional variables, it was the same. No statistically significant correlation between burnout and that type of hospital, specialty or radiology specialization, residency program, wards or shifts, seniority, provision of work outside the normal program level, or economic income, that's very interesting, or replacement of staff during holiday periods. So there is no statistically significant correlation between burnout and socio-demographic or professional characteristic of respondents and the presence of burnout syndrome before and during the pandemic. So these are the results. The most of the studies are made in the USA and Canada, because for the moment, I didn't find it in other countries. So you can see that the emotional exhaustion are more than a half in every radiology. So that in these studies, the feeling of depersonalization, a lack of empathy, it was the same, almost at the half. But the personal achievement in the Spanish radiologies, more than a half of them felt that they don't have a good personal achievement. These are interesting numbers, and I think that shows us that the burnout is more than outside factors, and this is an inside factor, no? So the third study of my PhD, it was an online survey just for the radiologists in Hospital La Fe de Valencia. And it was divided in two periods. First, during the second weekend of February 2021, and the second time during the last week of May 2021. Because in that period, we had a very, very high number of COVID diseases, because you know that in Spain, it was very affected all over the world. And the main cause of the radiologists that associated the burnout, it was with isolation at work. So that speaks about the importance of the communication, no? So the primary causes of burnout were the night shifts, the chaotic work, the loneliness or isolation at work, the lack of family time, no? It's so important to be with our family and friends, the failure of computer or devices that are directly involved in our work. So some factors that we can use to prevent burnout is the organization. And this is for the two levels, that the organization can speak with us and the organization in our work now. So when we start our work, we can organize and say, okay, this is where we would like to do, this is what is my responsibility, and it would be very, very nice to do it. The communication is fundamental for the people, I think, independent of the religion, the language, and the culture. Communication, it's less used because we have to create environments when we can express ourselves and we can ask questions without fear of being judged. And of course, for the doctors, we have that the problem of hours of sleep, no? Because our job is very demanding. We work a lot of hours per week, and afterwards we arrive home, and sometimes we have to work for our job. So, and the most important thing is to have a social life, to see our family, to see our friends, and to feel that we have a support, no? Because the human being, it needs to be felt understand, no? And to share their problems. And doctors, normally we have a lot of ego and we don't share our problems. So normally we isolate ourselves because we cannot communicate with other people, because if we are seen like less so fragile, no? If we are seen less powerful, so the people can misjudge us. So it's a thing that we can work on it, I think, of all over the world. And the hobbies, I think this is the most important one, because normally medicine is so absorbent, and sometimes we forget to do the things that we enjoy and the things that we love. So these are the articles that are like the resume of my PhD. If you want to read it, please, it will be like just 3-4 minutes. And I would like to end giving you these two of Albert Einstein. I think that's very important, because we cannot solve problems by using the same kind of thinking we used when we created them. And no problem can be solved from the same level of consciousness that created it. I hope that this is to help you to think about your situation, because I really think that we are connected, and it doesn't matter from where we are. At the end, we have the same problems. We want us to be accepted, to be loved, and to work together. So thank you very much for your attention. And if you want to contact me, I will gladly work with you. I think this is amazing to be here. And I'm very grateful for all of you and to sharing your knowledge. Thank you very much. Okay, so being said that, we are going to go with the next presentation. I will stop sharing screen. Okay.
From Shandong University, China, and is entitled Further Development of Near-Infrared Mediated Quantum Dots and Paclitaxel Correlated Nanostructured Carrier System for Cancer Diagnostic. On the introduction, I'm going to give you the background of the study. Research contents is what has been investigated with regard to the research work. And then conclusions and innovations, what conclusions has been drawn and innovations made. The list of publications is basically publications that were made from that research work. So they will be revealed, so that at the end of this presentation, you will be able to access them and be able to know details of what we've just discussed. Acknowledgements, just a vote of appreciation, where I would give my profound gratitude. Then questions and answers, I'll expect my colleagues, this is an opportunity for my colleagues to ask questions, which would be swiftly answered. All right. Now, worldwide, cancer is the leading cause of death. Hepatocellular carcinoma, HCC, which is a solid tumor, is the third and the ninth common cause of cancer-related mortality in men and women, respectively. Currently, with treatments of surgical intervention and chemotherapy, chemotherapy remains the larger portion of HCC treatment. The high prevalence of hepatitis B virus and hepatitis C virus predisposes one to develop chronic liver disease and subsequently develop HCC. HCC occurs predominantly in patients with underlying conditions, mostly chronic liver disease and cirrhosis. Once the tumor has been established at a particular location, it tends to expand. Now, the next stage will then have it where it will then spread within the liver, and then followed by tumor being spread across organs, which is referred to as metastasis, and ultimately resulting in death. So for this reason, there's a need for early diagnosis because there are different stages of cancer progression. And not only that, we should also have individualized therapy, because different patients tend to have different responses to cancer treatment. Diagnostic. The term there has got two main agents, as the name suggests. You have what you call diagnostic agent, right? Which is kind of a therapeutic agent. And then you also have what you call diagnostic agent, right? So the therapeutic agent is the one that gives molecular therapy. And then the diagnostic agent is the one that gives molecular diagnosis, right? This is what diagnostic term, the way diagnosis is all about, right? But then for this system that we created, what was the benefits of having a diagnostic system? One is that one would be able to assess biodistribution without having to invade the body. Secondly, you'll be able to see drug delivery as well as drug release. And lastly, real-time monitoring of therapeutic response, right? So again, this is critical for early diagnosis and individualized therapy if we were to win in cancer healing. Now, what strategy was it in place for this therapeutic system? First, the drug, which is a therapeutic agent, and the imaging agent, both have to be delivered to the target site or the cell or the target tissue. All right. And then also what was important is that the sensitivity of the diagnostic agent is very important. So that has to be increased for easy detection. All right. And then the therapeutic agent, the drug, concentration of the drug must be increased at the target site, right? So, and then overall, there must be low toxicity to the normal cells, because we don't want to hurt the patient. So normal cells are so crucial. We don't want to disturb them for the well-being of the patient. So we are only targeting the tumors. All right. Now, the nanoparticles. What are nanoparticle systems that are available so far? We have micelles, liposomes, dendrimers, solid lipid nanoparticles, and second generation solid lipid second generation of solid lipid nanoparticles called nanostructured lipid carriers. Now, one would have probably asked, why would I had chosen nanostructured lipid carriers? Because of two reasons. One is that nanostructured lipid carriers, they tend to have improved drug loading compared to solid lipid nanoparticles. Two, there is improved stability. All right. Now, what are the benefits of selecting these NLCs? There's low toxicity. The particulate matrix are easily biodegradable and therefore resulting in non-toxic degradation products. All right. So the ability to incorporate both the lipophilic and hydrophilic drugs. Then suitability for drug targeting. And then because of large surface, it allows you to modify it. an easy production process, and the possibility of large-scale production. And lastly, cheap excipients, i.e. lipids and stabilizers. What are the materials that we use in these non-structured lipid carriers that we developed? Glycerol monosterate. It has got a emulsifying character and forms a rich surfactant film. And then we also had SPC, soya phosphatidylcholine or lecithin. This one has got emulsifying property as well, just like GMS. But another very important part of it is that once we have it, then you'll be able to easily solubilize polysoluble drugs. All right. Then oleic acid. Oleic acid has got a low melting point, and this is very important because one, it increases mobility and fluidity of the surfactant layer, and also it promotes small particles. Remember, we are dealing with nanoparticles, so it's important that we should be able to control the particle size. All right, and then polonic F68. This was a surfactant, and the good thing with this is that it enabled control release, and then also allows stability at higher temperatures. Now, the PTX, you're all familiar with it. It's a very old drug. This drug was approved by USA FDA, Food and Drug Administration, in 1992 as an anti-tumor drug. It's a drug of choice for treatment of human cancers, including HCC. Now, what happens is that the mechanism of action of paclitaxel or PTX, in brief, it binds to heterodimers, alpha-beta heterodimers, and also it acts as a stabilizing agent for microtubule stabilizing agent. How does it do that? How does it do that? It inhibits what you call the microtubule dynamic stability, and then this will then what it also does is that it enhances polymerization, resulting in mitotic arrest, which then ultimately lead to apoptosis. Now, PTX structure, look at PTX structure there, you'll see that the structure is fused and bulky. This accounts for the high lattice energy of PTX, and also that if you look at the structure again, you'll see that it has got lipophilic species, and these they contribute to the very little, very limited kind of aqueous solubility of PTX, right? Now, FDA also approved in 2013 first line of treatment for adenocarcinoma of the pancreas, the PTX, and we also have PTX albumin-bound non-suspension, which is common in health facilities. Package size is 130 nanometer. So and it exists as an amorphous state, so it's non-crystalline. And also this apraxane is the color is from white to yellow, and it's kind of in a sterile lyophilized powder. And each single use vial contains 100 milligrams of PTX bound to 900 milligrams of human albumin. These things you are familiar with. Just give you a background of what is this about. remind you of them. So each ml of reconstituted suspension contains 5 milligram PTX formulated as albumin-bound particles. So you can see this is the box of that of that clinical formulation that you're familiar with. Okay. Now, what are the side effects of Apraxane? You have fever, chills, body aches, flu symptoms, sores in mouth and throat, hot and dry skin, heavy sweat, and feeling thirsty and even unable to urinate. So we also have unusual bleeding, especially in the nose, mouth, and hot tongue. All right. Now, I talked of therapeutic agent, PTX. Then the next thing that I will talk about is the imaging agent, right? Or which is the quantum dots. Now, quantum dots, why did I not take dyes, right? Now, if you had to compare dyes and quantum dots, you'll find that quantum dots basically have got superior properties. Look at this sensitivity. So remember we said from the previous slide, I had said that you need to have increased sensitivity of what? Of the imaging agent. So in this case, sensitivity, you can see quantum dots is superior to dyes. Then fluorescence lifetime 10 to 100 nanoseconds as opposed to 1 to 10 nanoseconds of dyes. So again, quantum dots have a superior property there. Quantum yield. So this is why then we picked quantum dots instead of dyes. Okay. Now, the design of the study, right, or of the formulation. So we have PTX, we have quantum dots, and the other lipid matrix, right? Now we had this quantum dots. What type of quantum dots did we use? CDTE. They've got what you call excellent optical properties. One, what I do know about them is that they have bright photoluminescence, and two, they prevent photobleaching. And also, we could also see here that we had zinc sulfate as a capping agent for several reasons. One, zinc sulfate increases fluorescent yield, quantum yield, from 5 to 15% to 30 to 50%. And also, it prevents oxidation. Thirdly, it reduces cytotoxicity. And fourthly, it enhances colloidal stability of the nanoparticles. All right. So the lipid matrix, oleic acid, glycylmonosterate, or soya phosphatidyl choline, which is this thing. So these are the ones that we used. And what is the rationale behind using those of that design? So the lipid matrix would provide protection from degradation of both the PTX and the quantum dots prior to their distinct size, right? And then the oleic acid, glycylmonosterate, and phosphatidyl choline have a drug status. In other words, they are generally regarded as safe. So there's no toxicity there, okay? And also formulation delivery, how it's going to be delivered. It's passive targeting. So we were aiming at that. We're using EPR, enhanced permeability and retention effect, right, of the vasculature, right? Now go into the research content. What was it that was done? Okay, so the particle size, so physicochemical characterizations, the particle size, poly dispersity index, and zeta potential. You can see that the correlated NLC, the particle size is 115.93 nanometer, as opposed to blank NLC. This is obviously this makes sense because this just track more compared to the blank NLC. So that's really good, right? And also, what we should know is that for the drug to reach the desired site, the particle size must be below 200 nanometer if we were to use EPR effects, right? As a method of drug delivery, right? Passive targeting. And also, it should range between, but though below 150, maybe below 200, like, but the best would be less than 150, all right? So polyspecificity index 0.17. There, then you can see that you get the Gaussian distribution. All right. So the morphology there, the black NLC, you can see the colored NLC, the particles. There's no aggregation. Also, you can see that the particles are smooth and they are nearly spherical. So that's good news, right? And then the encapsulation efficacy and drug loading. Right? Remember, we said the reason why we chose non-essential lipid carrier as opposed to showing lipid nanoparticles, SLN, is that NLC has got the advantage of improved drug loading. So you can see there is 4.68 there. Then encapsulation efficacy is good there, 80.70%, right? So there we didn't have to have quantum dots there because there was all wanted just to see the extent of PTX that can be contained in that formulation. Okay, so the high EE is attributed to colloid NLC's high partition coefficient and retention in organic phase as the nanoparticles solidify. Right now, then go into stability studies. Right. So if you look at the study there, you see that we used particle size and encapsulation efficacy over 4 weeks, and the particle size almost remained the same. So it's true with encapsulation efficacy. So suggesting that the formulation was stable over a period of 4 weeks. Wonderful. Now, drug release study. We had Taxol and the formulation colloid NLC. Please note this, that this is a biphasic pattern of drug release, in the sense that at the start there you have what called initial best drug release, and then secondly you have what you call prolonged drug release. And what you should also pick here is that below here is colloid NLC, and then above it is Taxol. This means that at any time point in the study, if you were to calculate the release of, say, Taxol and colloid NLC at the same time, you will find that Taxol would have, PTX from Taxol would have been released more compared to colloid NLC. This means then that the drug would take longer to be released with colloid NLC compared to Taxol. This means then that colloid NLC had superior drug release properties over Taxol. All right. Now, the MTT assay, cytotoxicity study, we need to see if within the cell there is toxicity exerted by this formulation. So we compared, we had black NLC, we had Taxol, we had, and then cell lines we used there was FG2 cell line, right? So you can see that the increased concentration is 7% to the is inversely proportional to the inhibition. So he said IC50 there is 1.05 for colloid NLC, whereas Taxol is 0.07. So this is in the good in the right direction. Okay. Now the cellular uptake study, right? We had MCF7 and HG2 cells, right? So the nucleus of the HG2 cells, the nuclei for both cells was stained with blue, and then for colloid NLC was stained with red, right? Now we use what call the DNA specific agent #3342. So, and you can see that compared to MCA, compared to HG2, MCF7 cells. You can see meshed here, this purple color tells you that they reached what? The nucleus, right? And what is interesting is that the MCF7, there you see more purple, suggesting that indeed the colony reached what? The nucleus. Now, this tells you something, that now this is not by mistake, because you remember that PTX, or paclitaxel, has been approved by FDA, the first line of treatment for breast cancer. So it is not amazing. This is just to buttress what we already know. So which is good, suggesting that the research is going in the right direction. Okay. Therapeutic efficacy evaluation. So you can see that we had normal saline, Bengal C, Taxol, and colon C. What we see here is that below here is the colloidal non-C, which is the formulation. Now, look here, you can see that it was very effective in retarding the tumor growth compared to Taxol, right? Taxol had a modest kind of, so meaning that it was significant, right? Taxol compared to the colloidal C, so it was very effective in retarding the tumor growth, right? And then the in vivo and ex vivo studies. So you can see that the arrow here, the arrow here indicates the direction of tumor, right? So you can see that the formulation went straight to where the tumor is, right? This is what we've been, from day one looking forward to. So you can see that our formulation is able to, so it was spot on. So we're able to get formulation to where we want it to be. So when you went straight to the tumor. Now, the fluorescence intensity there measured during that time period of 1, 2, 5, and 8 hours has been captured. You will see that fluorescence intensity decreased over time, right? Then after 8 hours, the mice were then sacrificed, and you see our tumor, the formulation is now here at the liver. You know why is the liver? Remember, this is our method of drug delivery was what? Passive targeting. Okay, so this means then that we are not prone to what? To RES, particularly in the system agents like in the liver. So the macrophages, the scavengers of the RES, we're not prone to them. So that's why we have a formulation after 8 hours being distinct to the liver. So it's because instead of having passive, rather instead of having active targeting, we had passive targeting. So this actually is something in the passive action. Then it tells you that the next step to do after this would be to do active targeting, because we now know that through this study, we've been able to develop a diagnostic system that is able to get the drug and to the tumor, and also we'll be able to detect the tumor without having to invade the body. So this opens another case for research. Active targeting would be at least we now know that it's working, right? And then histopathological assessment. So you have normal saline, Bengal C, and colon C. You can see that the biopsy by inspection, they are fairly the same, suggesting that there's no toxicity, and so there's no necrosis. All right. And on conclusion, you can say that we can safely conclude that the colon C formulation may be potentially an effective diagnostic translation system. So this is based on what we have discussed, the biphasic pattern that was that I did talk of, demonstration of the cytotoxicity, and then the MC-F7 internalization of the coherency, and then short-term stability, and the tumor, the colony NSC ability to retard tumor growth. Then imaging was spot on as well, and then substantial degree of safety in terms of the histopathological assessment. So what innovations have been made? One is that we've been able to develop a diagnostic system that's PTX and quantum dots. And then secondly, this was effective in anti-tumor activity. It demonstrated that reduction in tumor growth. And secondly, we also had it was specific, so you were able to see the formulation going straight to where the tumor is. Okay. Right. Now, these are the papers that you would during your own time you would access or visit, so that you can have the details of what I've just discussed. All right. So feel free to contact me on that, because I think there are a lot of work that can be done from here going forward. So you can now know that the formulation can be good for breast cancer, and what we need to do is just active targeting, right? So that's something that we can develop from there. All right. Just want to thank those that sponsored this conference, and also I want to thank the organizers, and also the opportunity that they gave me to be part of this. Thank you so much. And at this juncture, I would now welcome those with questions, if there are any.
Yes. May I please? Hello.
Yes.
Can you hear me?
Yes, I can hear you.
Welcome to your presentation of the results of medicinal plants in the treatment of breast cancer. Breast cancer remains on the leading cause of family mortality, worldwide, driving the relentless nature of more effective and less invasive treatment options. In the context, we are exploring the facets and perspective of utilizing compounds derived from medicinal plants, offering a promising and innovative approach, drawing with delving the therapeutic properties of compounds such as curcumin, lopiol, quercetin, and the alkaloids, examining their potential impact of breast cancer treatment. Let's explore the possibilities of significant advancements in medicine, these devastating diseases, checking the outcome for patients. Let's embark in one journey targets. Relevance of breast cancer as global health causes of mortality, urgency for more effective and levels challenges in treatment. In the essence, and the captive concept of utilizing medicinal plants as promising solutions in the treatment of breast cancer, the inherent qualities of these plants offer is unique and potential alternative to conventional approach researcher. Studies have illuminated its noteworthy anti-cancer potential, showcasing its ability to impede cell progression and stimulate apoptosis specifically in breast cancer cells. I will help delve into properties of luteal present across diverse botanical sources. It is how it is arresting in the advancement of cancer cells and the fostering programmatic cells. They underscores it's significant in the pursuit of effective. Quercetin, a potent flavonoid abundant in various fruits and vegetables, unfolds as a noteworthy contributor to cancer effects. Extensive research has revealed in this capability to induce apoptosis. The programmatic cell is crucial for mental health cellular balance. Furthermore, quercetin plays a pivotal role in inhibiting cell proliferation, underlying the under-controlled growth characteristics of cancer cells. In the exploration of quercetin properties, derived from nature's bounty when uncovering this potential as available components in the pursuit of cancer strategies. Researcher was working on several fronts to explore the range of natural products where available mainly from the Amazon region in their chemical properties and potential combat cancer cells. Other studies are recorded from the collection of material and extraction of chemical compounds to in vitro tests and clinical studies. The results are encouraging and the potential important for consideration medicinal palms as important adjuvants in the fight against breast cancer. The use of medicinal plants and their active constituents represents our hope food and the novel direction in the search for alternative therapies for breast cancer with potential for significant advances in managing the devastating disease and improving patients' outcome. Thanks and recognition, universities of Gurupi, University Brazilian, a concert regional pharmacy Tocantins. Thank you very much.
Excuse me. May
A hearty welcome to all of you being interested in those exogenic risk factors for cancer contributing to failure of natural resistance when autonomous regulation of defense functions are damaged by unwanted and irreparable neurotoxic effects, making the toxically affected central nervous system dysfunctional, especially the autonomous functions. In particular, the irreversible neurotoxic effects on the autonomous functions as part of the polyneuropathy are often confused with endogenic psychiatric or so-called psychiatric depression, whereas the exogenic factors rather involuntary caused deficits by external causes, including a tendency to evolve into suicidal ideas, finally precocious death. The clinical routine neurological diagnostics are clearly missing the relevant neuropsychological extent of intoxications supposed to be reversible in the majority of cases but develop irreversible effects in the long run, marked as latency period. The epidemiologic approach of defining and depicting exogenic risk factors as to consider even requires a lot of methodological aspects to be applied for good practice. In fact, for any successful prevention of long-lasting persisting effects, the crucial quantification of a dose-effect relationship is a must for trustworthy, mindful, and helpful contributions of effective prevention. As soon as environmental risk factors for breast cancer, in particular with unusually early age groups, have been defined, the urgent dimensions of their specific causal contribution may require epidemiologic results which are repeated in several aspects. The example of discovery of risk factors for cancer in male chemical workers to this end, with numerous reports of established occupational risk factors, were strengthened by the statistical analysis of unplanned natural experiments in humans, mostly male populations, which occur even next to each other, living in a chemical world. Animal experiments are usually well planned and observed over short periods without developing cancer, because they are killed before the cancers develop. The opportunity to evaluate unplanned human experiments has drawn attention to the ubiquitous chemical hazards during life expectancy. This key presentation points to the relationship of the onset of early signs and clinical manifestation of breast cancer by looking at the chain of events from the viewpoint of environmental, early direct toxic effects on the brain with short latency for malignant growth, such as cancers affecting the target organs, as mentioned already, to be endocrinically stimulated glandular structures of the breast. The counting unit, nevertheless, is a lethal outcome for worst-case deceased. Especially the neurotoxic exposure-effect relationship before the precancerous carcinogenic effects in males, with latencies between neurotoxic symptoms such as sexual deficitary functions and loss of libido in young age groups below 35 years, is becoming of age. That is well known. But starting with vomiting, loss of consciousness, nausea, and loss of cognitive abilities, as well as irreversible behavioral changes defined by WHO as neurotoxic encephalopathy. One approach to pinpoint a risk to a cause is related to the misinterpreted claim that so-called low doses must be harmless as concerns the immediate damage of chemical substances as soon as irreversible effects are observed in the long run. Mostly known in this category are polychlorinated carbohydrates, shortcut name dioxins, as well as some biphenylic PCBs, altogether proven to cause dioxin-related effects, which are concerned. These are undetected byproducts in any synthesis-based processes during the production. They are unavoidably contaminating the process as unwanted toxic substances of the de novo synthesis, not easily preventable, let alone when combustion of garbage, such as plastic material, with temperatures around 400 degrees Celsius, as it may occur not infrequently in these days, as long as it is not taken as a serious problem and condemned. Those polychlorinated dioxin congeners as part of the dirty dozen are hidden neurotoxically damaging products. Tetra, CTD or TCDD, penta, hexa, hepta, and octachlordibenzodioxins. According to the context of this conference itself, these introductory remarks are illustrated by two stories. The first was happening in Italy's southern region around the Vesuvius. The second in a German chemical plant. One additional opening remark: the combustion of plastics in open air without protective measures is able to contribute to the toxic air pollution when unshielded as required by incinerators. The consequences may be irrefutable. This topic is part of the scientific approach, which is called observational studies by analytical epidemiology, and it serves pointing to a public prevention problem solving effort and is not unimportant, especially if criminal aspects prevail, whereas the production of herbicides and biocides from chlorinated products has been promoted without control for adverse effects. As material to get hexachlorocyclohexane or lindane are some of the dirty dozen products, such as biphenyls, disclosed as a risk factor for cancer. These are found to be associated to increased breast cancer rates in those women which were under risk in young ages and altogether even selected for health when employed in a chemical factory. That plot of the second story of a high risk in young age groups and epidemiologic ongoing cohort study shall serve as an example of quantification of cause-effect relationship in young women. Story 1 is the observation around the area where the Inferno of Calvi Risorta near Naples occurred. At the time of unrest in the press, the publication of Rosato et al. Related the metabolic syndrome as very likely to be associated with unexpected early premenopausal breast cancer risk, applying different index categories. But the question remained open: what causes the metabolic syndrome at the beginning? The two related publications by Capasso et al. Offered interesting findings but had different aims. In 2010, the National Cancer Institute of Naples published a report by Capasso et al. With co-author Maurizio Montella, suggesting that several evidences indicate an association between metabolic syndrome and breast cancer, primarily owing to insulin resistance and low serum HDL-C. But also referring to an American study by Cabot et al., the co-authors drew attention to these findings on the issue of metabolic syndrome and breast cancer risk as already addressed, but the question was left open. The women's own decision to eat the wrong food would be preventive proof? The conclusion of Capasso et al. Was in time-dependent, covariate analyses. However, certain scenarios indicated a positive association between the metabolic syndrome and breast cancer due primarily to positive associations with serum glucose, serum triglycerides, and diastolic blood pressure. These were only symptoms of a syndrome, neither exciting news nor hinting to the causes. The Italian author suggested, the National Cancer Institute of Naples has pointed out the association of the metabolic syndrome and breast cancer risk in postmenopausal women. Premenopausal women have not been included in the study group, but neither female industry workers. Concluded was that metabolic syndrome features before cancer incidence presents an indicator of breast cancer risk. The unsettlement of the hormonal arrangement in postmenopausal women, along with an increased obesity, probably favoring the hormone-dependent cell proliferation for tumorigenesis. Conclusion of the authors: adjustment in lifestyle in postmenopause. Query: in the region of Naples, with the notorious garbage combustion problems, how to change nutritional elements as the only advice or preventive action? The mention of unsettlement in the text needs explanation in what was the cause of giving it a prominent position in the conclusions, instead of addressing the metabolic syndrome as an indicator of urgent recommendations and swift action to enhance and warning to consume toxic products from their fields. At the Warsaw Conference on Cancer in 2013, one of the Italian authors was scheduled to present the data and their relationship to a direct urgent environmental problem in Naples and the indication of a causal role. Before the meeting, I had contacted the Naples research group by a letter to express my hope to learn more by discussing the issue of air pollution by the Inferno of Calvi Resort near Naples, which had been blamed in the press. The speaker for the topic, Maurizio Montella, however, he did not show up at the conference, alas. To relate the garbage incineration problems around Naples, especially plastics, but also other chemical residues, was somehow noteworthy because the air pollution with dioxins such as the Seveso dioxin TCDD by organized crime in the Vesuvius region has been rather disturbing for the entire population at that point in time. The recommendations to women to adjust their lifestyle was out of place since numerous press reports of dangerous environmental exposure to pyrolysis products such as the notorious garbage disposal problems resembling ashes raining from the volcano had been related in the press to the toxic waste inferno of Calvi Resort in the smashing area of Naples. The second Italian publication, however, avoided this discovery by focusing to the variations of metabolic functions in the population and individuals. The title, Metabolic Syndrome, Breast Cancer, Link, varies by intrinsic molecular subtype by Capasso et al. In 2014 to appear related to Capasso's paper from 2010 did, though not pick up the new extreme focus to the individual men or women, but describe the frequencies of pointing to individually susceptible per person, thus unavoidable to live under risk. It is the meaning perhaps may be unfit for the environment where they lived in, with view to the first report in 2010. In the contrary, the message as published was changed to new molecular biomarkers unveiling metabolic syndrome-related breast cancer carcinogenesis need to be detected to further stratify breast cancer risk by subtypes, and subtypes are the individuals. It did not consider findings in a German factory already published since 1995. This comes to story 2. This contrast refers to a scientific approach by following up males and females in a German chemical plant manufacturing those biocides from chlorinated carbohydrate compounds containing dioxin congeners, ever since the invention of the herbicide lindane and others such as Agent Orange, useful for defoliating one half of Vietnam in the 70s. The workforce had been followed up for cancer mortality risk estimation by a cohort study since the closure of the entire production site 1984 and published 1995 in international reports. Thus, from the beginning of attention to the higher risk ratios for cancer and suicide as main causes of death for far over expectation, it was an open question why the cancer appeared only much later than those exposures with respect to the decreasing half-time of the congeners. It was the time to look at acute and subacute symptoms after workplace exposures in the medical direction, focusing on psychoorganic syndrome, the internationally POS-related irreversible signs of persistent toxic encephalopathy. The mortality has been presented for males and females by groups of causes of death, having been particularly specific for causal association with TCDD exposure at the workplace. For males, the overall mortality was significantly increased, and also for malignant tumors of the airways, not however for malignant prostatic or pancreatic glands due to the small observed numbers. Suicides were significantly increased too. The table shows the causes of death by standardized mortality ratios, which are related to the expected rates of male mortality ratios in the population of Hamburg. The main causes of death were the cardiovascular diseases, malignant neoplasms, and then upper respiratory organs cancer, including the upper respiratory organs, and the total mortality significantly increased above expectation. For the female workers, the same rates were shown for 390 women. The cardiovascular disease significantly lower than expectation. Malignant neoplasms and cancer of upper respiratory organs somehow increased but not significant, whereas breast cancer is clearly significantly increased by a doubling of the deaths compared to the expected numbers. The total mortality also was below expectation, which was insofar important as these effects show very clearly the very high susceptibility for breast cancer or even tumors of the glandular organs. The deaths from unnatural and undefined causes showed a significantly increased association to SMR 2.95, so a threefold increased risk, suspicious to contain a great number of suicides, which were not always specified enough because of religious constraints as self-inflicted cause of death. This manco has to be mentioned, though, because the close relationship to the irreversible central nervous affections due to the toxic effects of endless complaints and unbearable behavioral problems during many years. The US Army veterans, having been deployed to Vietnam, had developed similar lifelong psychotoxic effects and cancer risk increases. Army veterans have been also the reason that the research was intensified. Final remarks. The mortality of entire cohort of 1,589 German workers, evaluation of cohort effects differed in that the overall causes of death were all significantly increased for males in the production, whereas for the subcohort of 390 women, however, only the breast cancer risk was statistically significantly increased by contributions from the younger lethal cases exclusively. This was contrasted with the decreased overall female mortality, indicating a healthy worker effect, a strong indication of occupational risk factors being identified as monocausal because they were popping out of the normally low mortality as a cause of death. The plant had to be closed and dismantled in 1984. The results were published in the time period starting 10 years later. For men, distinctly two malignancies of the lung and respiratory organs, as well as prostate cancer, were associated with suspicious lethality substantially increased above expectation, irrespective of statistical significance for prostate. The prostate is one secreting organ, endocrinologically regulated by the autonomous centers of the male brain, as well as the glandular part of the mammary gland in women by stimulation of estrogen and prolactin. It is noteworthy that women aged 15 to 64 had shown an increased breast cancer mortality, especially in the age group 45 to 49, with standardized mortality rate SMR 7, sevenfold increased risk to die of breast cancer. Not included are singular surviving cases after successful treatment still being followed up until now. An additional evaluation based on an unpublished document contains the figures presented with details for breast cancer, covering the follow-up period from 1955 until 2007. Mortality for breast cancer after different duration of periods with TCD exposure risk as a female workplace had been up to 1984, where the duration of exposure below 1 year, SMR 184, 5 observed and 2.7 expected. So the 1 to 10 years exposure had a rate of 1.16, 6 over 5. But 10 years of exposure had the rate of 3.28 and strongly significant as a more than threefold risk as an occupational disease. Fact is the rising of the risk estimates by exposure longer than 10 years during early years of production without any prevention depicts a very strong hint at the causal occupational risk due to TCDD. The follow-up is continued until 2023, but incomplete as to this date, whereas the results are confirmed after 16 years, apparently not influenced by survival rates from breast cancer due to medical measures. Thank you very much for your attention. Wishing you a pleasant and successful conference. Goodbye.
Honorable assistants, allow me to start my presentation entitled Recalcitrant Paraneoplastic Pemphigus Associated with Prostate Adenocarcinoma, Spectacular Evolution under Androgen Deprivation, as a collaboration between the Department of Dermatology and the Department of Oncology and Radiotherapy of Ibn Rushd University Hospital, Casablanca, Morocco. We have no conflict of interest. Paraneoplastic pemphigus is a rare life-threatening condition associated with polymorphic mucocutaneous manifestation with an underlying neoplasia, mainly represented by hematological malignancies such as chronic lymphocytic leukemia, non-Hodgkin's lymphoma, Castleman disease, and thymoma. Paraneoplastic pemphigus associated with prostate adenocarcinoma is an unusual situation. It's about a 69-year-old patient with no medical antecedent or family history of malignancy or autoimmunity, presented a severe oral erosive mucositis evolving for 4 months. So, physical examination showed stomatitis and crusting lesions of the lips, marked conjunctivitis, endonasal erosions, and some cutaneous and some polymorphic cutaneous erosions of the neck and the upper trunk. The Nikolsky sign was positive. Skin biopsy, direct and indirect immunofluorescence was confirmed the diagnosis of autoimmune bullous disease type pemphigus vulgaris. So the patient was treated by prednisone 75 mg per day and azathioprine 150 mg per day. No improvement was noted after 1 month of immunosuppressive therapy. Considering the polymorphic mucocutaneous involvement, the severe stomatitis, and the no improvement after 1 month of conventional immunosuppressive therapy, a paraneoplastic pemphigus was suspected. Serum tumor markers markers including prostate-specific antigen, alpha-photoprotein, CA19.9 and CA125 were negative, except for an elevated level of prostatic-specific antigen. The PSA was elevated at 23.28 nanogram per milliliter. So the patient underwent an abdominal and pelvic echography showing a prostatic hypertrophy with calcification. So to better access the prostatic volume, a multiparametric prostatic MRI was performed showing 2 prostatic lesions in the transitional zone, classified Pirates 5, and 1 prostatic lesion in the peripheral zone, classified Pirates 4. So a prostatic biopsy confirmed the diagnosis of a conventional prostatic adenocarcinoma with a Gleason score 8, 4+4. Thoracic, abdominal and pelvic computed tomography and bone imaging showed no significant abnormalities. So the patient was classified as a localized high risk, and a treatment based on hormonotherapy was recommended for 18 months. The patient was treated with subcutaneous injection of goserelin. So the subcutaneous injection of goserelin was at 10.8 milligram per 3 months. And after only 1 week of the first injection of hormonotherapy, a spectacular regression of mucocutaneous erosions was noted, with no recurrence of dermatological symptomatology after 1 year follow-up. Radiotherapy on prostate, seminal vesicles, and lymph nodes was also indicated in our patients. So let's move to the discussion. Prostate cancer is the second most common urological malignancy associated with paraneoplastic syndromes after renal cell carcinoma. However, paraneoplastic pemphigus associated with prostate cancer is exceptional. To date, only 2 cases are reported in literature. So the etiopathogenesis of paraneoplastic pemphigus is not fully understood. Skin lesions may be the result of an autoimmune response generated by antibodies directed against tumor antigens that cross-react with epithelial antigens. For the clinical features, stomatitis is the earliest and the most constant sign of paraneoplastic pemphigus. The cutaneous lesions are polymorphous. Histological criteria are non-specific, and the treatment is based on the reduction of autoantibody productions. So immunosuppressive therapy should be administered promptly, and the first-line treatment is systemic corticosteroids associated to corticosteroid-sparing agents such as azathioprine, mycophenolate mofetil, and cyclosporine A. So the rituximab with or without intravenous immunoglobulin can be an interesting therapeutic option in the case of the failure of the first-line treatment. So it is very important to treat neoplasia associated with paraneoplastic pemphigus. In our case, clinical improvement was observed only after combining hormonotherapy with conventional immunosuppressive therapy. So in conclusion, paraneoplastic pemphigus associated with prostatic adenocarcinoma is unusual. It is a challenging condition because its etiopathogenesis is not fully understood and because of the poor effectiveness of conventional immunosuppressive drugs. Androgen deprivation may be inefficient adjuvant therapy of paraneoplastic pemphigus in men presenting an underlying prostatic adenocarcinoma. Thank you for your attention.